most determined abiotic stresses, which critically damage the crop plants and cause significant reduction in yield of virus crops, particularly fruit crops, which cannot resist any, high salinity. If we're talking about adaptation strategy, all plants under abiotic stress conditions use virus. Sorry, Leo, I think some. Okay. So please, Mr. B, all other please mute her microphone because I hearing some kids' voice. Okay.
So, hello. Can I others mute your microphone, please?
Just mute your microphone.
So, apologies, Dr. Walid. I just muted them.
No problem. Okay.
Yeah. Okay.
Okay. This is another problem for mom who worked in science field. I know. Okay. Also, there's another factor I didn't mention yet is heat waves. Heat waves are projected to more intense and longer lasting and affect negatively on orchard or fruit tree, particularly during fruit set or in June drop in the next month, as we know. Heat waves could lose يعني major crop from any fruit trees, especially deciduous fruit trees, which يعني during the period of June drop, most of deciduous trees like grape, peach, apricot, almond, the crop was in the pre-final stage or pre-maturity stage. Heat waves affect negatively at the drop major percentage of this crop, and all farmers lost their major crop or economic crop, which remain in the tree just maybe 25% of total the crop after heat waves. I talked about this point because I think all scientists of plants, not only fruit scientists or orchard, attractive with this heat waves. Heat waves now become very critical for all plants, not only for orchard fruit. Okay. As we say, we will back to adaptation strategies. Plants used various adaptation mechanisms that include morphological, physiological, biochemical, and molecular translation in gene expression to avoid adverse effect of stress. Salinity and drought there is a correlation between drought and salinity. When drought increase, salinity of course increase consequently after drought. Due to drought, this mean there is no rain. Okay, plant absorb or uptake water from soil to complete the growth cycle. Salinity increase for drought, for excessive use of fertilizers and other agrochemicals. Okay. Drought and salinity considers the major climate threats for all plants and inhibit plants and lose their the plants lost their crop, maybe in dangerous phase. Plant could lose burned or lost the mean vegetative growth. To understand how these mechanisms are regulated and how to overcome the adverse effects of drought on plant productivity, we will discuss some adaptation mechanisms which used to enhance the quality and yield of the crop as for. First, we will discuss morphological adaptation or exposure morphological adaptation, which tree adapted to used to adapt to the abiotic stress in various ways. The evolutionary modification of root system canopy architecture, reducing leaf size, partially shading, or dropping new fruit set and small fruitlet, or other habits plant used to adapt it with abiotic stress. First, abscission of leaves in evergreen orchard like orange and other. Many crop had to shade the leaves partially when survive to water defect or drought for long time. Regular shading start with older leaves first to reduce water loss, then following young leaves. When droughts remain for a longer time, okay, maybe the tree is shedding the small fruitlets. Other strategy or other mechanism orchard use under the drought and other abiotic stresses, canopy architecture control. Canopy architecture play a significant role in adapting drought trees to stress condition. Currently, I think major breeder looking for dwarf cultivars in all kind of fruit trees with dense crown to be more tolerant for drought and stress and other abiotic stress. To avoid dehydration, tree dehydration under this stress through small crown trees and compacted and the small crown trees. Compacting canopy, reducing leaf and reducing leaf size, also change crown shape. All this factors reduce transpiration and save energy by prevent high light density, reaching the inner leaves and decrease penetration of the solar radiation. Also, dwarf cultivars reaching you reduce the sunlight reaching the inner leaves and decrease penetration of solar radiation to improve microclimate inside the tree. Therefore, dwarf cultivars, as we said, with compacting ground could tolerate the environmental stress more than open crowd varieties by delaying dehydration. Other mechanism, deep root system. The depth of root for any trees determine tolerant for drought because penetration roots mean ability to absorb more water from the soil. So tree use this mechanism under drought condition, particularly under severe drought condition. Some trees like jojoba shrubs or trees, it's one of drought trees. The root system reach up about 10 times double the canopy measurements. Furthermore, trees with deep root system like Robusta coffee has the better tolerant ability to drought conditions. Another strategy you plan to use anatomical adaptation through different parameters, as we discussed now. Under abiotic stress, trees use different anatomical adaptation to tolerate stress and survive through reducing water loss and closing stomata, which consider the main inductor for drought and different stress. Anatomical adaptation used to reduce water by closing stomata under drought stress to minimize Also, some plants minimize the number of stomata per leaves and the size of stomata to control water loss and use water available. Regarding physiological adaptation, there are different compounds discovered from maybe 2 decades only or 3 decades planted synthetic during abiotic stress to avoid dehydration. Biosynthetic of jasmonic acid, one of these substances, which of consider physiological mechanism to adapt it with stress condition. There is also plant could use different ways to synthetic jasmonic acid according to type of plastide cytoplasm or enzymes and peroxisome. Jasmonic acid has positive effects on salt tolerance. Currently, we use jasmonic acid as growth substances to increase tolerant under salinity stress or under drought stress. And there are numerous research about this point. Using jasmonic acid usually using as foliar application to increase tolerance of fruit trees, as we said, for abiotic stress. Also, it's improving vigor and vigor of tree and improve productivity. Also, under osmotic stress, plant activated different physiological mechanism to cope with reactive oxide solutes production by increasing organic solutes like glucose, malate, and proline to adjust osmotically pressure. For instance, under drought and high temperature condition, citrus trees, particularly that grafting on Cleopatra rootstock, activate biosynthesis of flavonoids to mitigate the oxidative effect and avoid damage during the drought stress. Also, from other side, breeding tolerant rootstock or tolerant type for abiotic stress, one measure strategy. Different countries looking for it now. As I said before, Cleopatra graft rootstock and citrus could tolerant drought. Other in apple, other in grape, there is other type. In most of fruit trees now, there is a type could tolerate this stress. Other point I want to talk about tolerant stress, role of potassium. Potassium, as we need, as we know, all know, potassium is a very important element for all plants, and this potassium considers the only element plant absorbed anyway when potassium available in soil. But any plant absorb potassium directly without limit any quantity. Cytoplasm potassium is very critical term of stress used to improve plant tolerant for virus stress. So under stress condition, particularly salinity or drought, plant cell lose potassium due to activation of case-based like endocellulose, which further lead to plant death. Potassium play, therefore, as we said, as I said, plant absorb potassium by any quantity from soil if available, or even used as a foliar application. Because potassium play as a charge balance in plant under certain condition, as invisible influx of sodium, which induce an approved membrane depolarization, which affect the transport of major essential elements across the plant membrane. Potassium retention in mesosphale cells in major strategy to existing of salt tolerance, therefore under soil salinity condition, fertilizing with adequate potassium reduce adverse effect of plant. So potassium is very critical element in plant mechanism. From my opinion, I think we must fertilizing any plant under environmental stress with adequate potassium to avoid to improve plant tolerance and survival under this condition. Managing practice, this which I mean agriculture practice use different orchard. Of course, there are different steps required to maintain orchard productivity under environmental condition, include proper irrigation, proper fertilization, maintain soil moisture, using rootstock type of rootstock tolerant for the drought or salinity or heat waves as the stress in the area. Also, exogenous treatments of plant growth substances like jasminic acid, as we said, or alcoholic sugars or other substances could be very beneficial under stress condition. Exogenous application to reduce adverse effect of plant stress. I make this in point to be easier and be easier to understanding. First application, full application of alcoholic sugar, as I said before, like sorbitol and mannitol. Now a lot of farmers and use sorbitol and mannitol to improve orchard tolerant and enhancing quality of fruits. Also using organic fusels like jasminic acid, salicylic acid, proline to increase tolerant for virus abiotic stress. Other application, I think, is commonly using in different orchard, humat compound. Humat compound in arid and semi-arid area is very, very important. Must use regularly during the season. I think currently we use in Arabian area or Middle East, we use 1 kilo or 1.5 kilogram per film humat per each tree. Okay, to improve tree growth and productivity. Also use proper type of fertilizers for the kind and for the physiological stage. As we know, we could use the different type of from fertilizer, from the nitrogen fertilizers, for example, in the growth season. Yani firstly, during we can say example about citrus tree. Firstly, if with growth cycle, spring growth cycle, we start to use sulfate ammonium, not nitrate. Okay. After that, we use nitrate. After June drop in July and August, we use again sulfate, ammonium sulfate. All this kind is just nitrogen fertilizer, but we use different type from it. Also, amino acids, consider another type of nitrogen fertilizers. We must take care during using amino acids if we're looking for storage fruits on the tree or elongate harvesting time. Also, calcium and potassium play important role in improve trait tolerant under drought condition, particularly during fruit set and fruit enlargement. So calcium, fertilizing with calcium even soil application or foliar application is very important and increase fruit quality. Another element, micro element like zinc and manganese is very important to improve tree growth and fruit quality. Growth regulators, another important factor currently used in program fertilizing program like gibberellic acid, G3 and cytokinin play important role in plant metabolism and particularly under stress condition to reducing stress on the plant and provide some substances trees need. Conclusion for our lecture today: under stress condition, plant change all metabolism to activate all processes and save energy metabolism along to leading the accumulation of photoprotective and antioxidants secondary metabolism and oriented to mitigate the damaging of different stress. The purpose of this metabolism and this adaptation retain a high photosynthesis activities and to cope to plant coping with oxidative stress and preventing the accumulation of antioxidant metabolism. Fruit trees particularly use different adaptation, as we said, strategy to cope with adverse effect of stressful condition and survive under environmental stress without loss of yield or productivity. Thank you very much. Any questions? Mr. Liu.
Yeah, thank you very much, Dr. Wali Fawad, for your nice presentation. So I would like to go for the participants. Do you have any queries to discuss with Dr. Fawad so that he may be able to answer your queries?
I'm waiting.
Of course, good morning to Professor Fawad. I am Dr. Kakuli Banerjee from India.
Good morning, Professor.
Yeah, I'm from Central University of Orissa, India. Actually, I wanted to have a very simple question. How does the mangroves in the salt stress condition, how do they overcome their physiological changes in terms of in the changing era of climate where the temperatures are very high? Do they have any specific physiological changes because of which the amount of carbon assimilation is decreasing or increasing, or there is some change? That is the question which I want to like you to know, focus on.
Mangrove as an anti-plant which growing on under high salinity, so growing on seawater, not on regular places. Mangrove has different physiological adaptation strategy to cope with this condition. Okay, mangrove have a lot of compound inside the cell to have to balance cell pressure. There is a lot of solutes, organic solute, compound solute inside the leaves. Also, mangrove have small leaves. As we said one strategy in canopy architecture control and dwarf types, smalling leaf size. Mangrove have a very small size, very small leaves, as you know. So transferation from the leaves is minimum if we compare with other kind of orchard like apple or trees or grapes. Okay, this means there's 2 point mangrove have to tolerate high salinity condition, have a lot of organic solutes and cell. Okay, Other have smallest leaves if we compare with other trees. Okay.
But in some cases, we do have broad-leaved mangroves. We do have broad leaves. I mean, is there any change in the photosystem too, which occurs when there is a lot of salt stress or a lot of temperature rise, that the physiology is hampered and the amount of photosynthesis that is supposed to take place is unable to occur? Is some case like that, or it is a normal phenomenon of mangroves to survive in stress conditions?
Mangrove normal surviving under high salinity condition, and if you plant it on the roof under normal condition, cannot grow. A mangrove tree must at least salinity condition not less than 10,000 ppm. If you add water tap, mangrove could first stop the growth, secondary it will die. This need high salinity condition. It's only grow on shore. Seashore, okay, mangrove grow in it. In Egypt, by the way, we have about 4 area on Red Sea growing mangrove naturally. Okay, on Red Sea, the east coast of Egypt.
It's a Bay of Bengal.
Yeah, okay, we have this but regarding photosynthetic, okay, as we know, there is photosynthetic and other night photosynthetic. Okay, mangrove have a strong photosynthetic system. Okay, to avoid photosynthetic during the day with high temperature and solar radiation. Okay, mangrove depend on night photosynthetic. Okay.
Yeah. Okay.
Okay.
Thank you, sir.
You're welcome, Professor. Any other question?
Yeah, thank you very much, Dr. Fowle, for your nice response to Dr. Kakoli Banerjee. So still any more queries? You're welcome. Or else if there are any queries, we will move on to the next presenter, Dr. Ivana Sola.
Yes, hello everybody. Regards from Croatia.
Dr. Ivana Sola, good morning. So before you start your presentation, so I would like to introduce a brief about starting your biography. It's okay? Yeah, okay. Thank you very much. Thank you. Dr. Ivana Sola works in laboratory for phytochemistry at the Division of Botany from Department of Biology, the Faculty of Science from University of Zagreb, Croatia. Her main scientific interest is plant-specialized metabolism plasticity. For example, changes in concentration of different metabolic synthesis of new compounds, genotype or phenotype adjustments, ecotype or yield respectively, or bioactivity and bioavailability changes, and possibilities like the use of a specialized metabolites profile as an indicator of stress in the environment, boosting of specific metabolites in order to produce a value-added plant food, and screening of specialized metabolism changes for prediction models. She has published 18 scientific papers so far and is a co-author of 1 manual. So far, she has participated as a collaborator in 4 bilateral German-Croatian projects, in 1 scientific project from Croatian Ministry of Science and Education, 1 from Croatian Academy of Science and Arts, and 1 from Croatian Ministry of Agriculture. Currently, she is leader of 1 Croatian Science Foundation project and a collaborator on another. She is a special issue editor in Journal of Plants and a member of several associations and societies related to plant biology. She teaches and holds practica in plant anatomy, plant bioactive substances, and microbiology of plants. Now, I would like to call upon Dr. Ivana Sala to present her presentation on improving the phytochemical profile and bioactivity of Chinese cabbage sprouts by interspecies transfer of metabodies. Dr. Ivana Sala, you're most welcome to start your presentation now. Thank you so much.
Okay, thank you. Just a moment.
Okay, take your time.
Yeah. Okay, thank you. Thank you for a nice presentation about my work. Just let me see here. Okay. Okay, can you all see
Yes,
the presentation now? Hopefully, yes.
Yes, we can see your screen.
Okay, great. So, okay, you can see the screen. Great. So, once more, regards from Croatia. My name is Ivana. I'm working in the lab for phytochemistry at the division of the Department of Biology, Faculty of Science, and in scope of this e-conference, I decided to present some of the results that we gathered in the last 2 years. So the title of my presentation is Improving the Phytochemical Profile and Bioactivity of Chinese Cabbage Sprouts by Interspecies Transfer of Metabolites. So how did we come to this idea? Here are some of the papers that were very interesting to us, and we decided to make some investigation further on. So a German group in couple of last years has published several papers which show that there is a possibility to change bioactive components between different plant species, and not just in terms of allelopathy, but as well in a term of some new phenomenon that they called horizontal natural product transfer. Namely, what they have seen is that if they grow plants like peppermint near the plants that contain nicotine, like Nicotiana tabacum or tabac, then the peppermint plants will also be enriched with this alkaloid nicotine. And then later on, they made some further investigations and they realized that interspecific transfer of alkaloids is an unconsidered source of contaminations in different plant species, and one should always analyze the phytochemical content of a certain species if it is grown next to another plant species, because they can actually change their chemicals. So they have been mainly working with the alkaloids, and then we decided to work further on, and we investigated whether this horizontal natural product transfer could be realized with other type of plants, plant compounds as well. So we concentrated on phenolics, mainly flavonoids and phenolic acids. And the question that we made to ourselves is, can the phytochemical transfer between different species affect their bioactivity as well? So not just their chemical profile, but their biological effects and even gene expressions. So what we decided to do is first to check the plant phenolic and vitamin C profile in plant donors and plant acceptor, and then check the gastrointestinal bioaccessibility of phenolics and vitamin C, the antioxidant capacity of plant acceptor, its hypoglycemic potential, anti-proliferative effect, and finally gene expression. So we were interested in whether the change in phytochemical profile could actually mean a chance for plant acceptor later on. So this is how we created our study. We decided to take Chinese cabbage sprouts as a model of plant acceptor. Sprouts are actually now very often used in the average human diet, especially here in Southeast Europe. In Croatia, you can find in our stores many of fruits. People use them as a fresh food, or they can even cook them, or just add to normal salad every day. Why do we eat them and love them? Because they're rich in bioactive components. They have 10 times higher concentration of some healthy bioactive components compared to the mature plant or adult stage of the plant. They are tasty, and one of the main advantages is that you can grow them very fastly. In 7 to 10 days, you can get a fresh mass that you can already consume. And of course, you don't have to have a soil substrate or a field. You can grow them on a wet filter paper or a wet cotton in your apartment or at your workplace very easily. So it's not time consuming, and you will get your fresh plant material, fresh food. So we took these Chinese cabbage sprouts as a plant acceptor and used 4 different plant species as plant donors of their bioactive components. So the whole study was designed like this. We prepared the water extracts of plant donors and then incubated Chinese cabbage sprouts or plant acceptor in these extracts for 4 hours. Later on, we have thorough seedlings and transplanted them onto the soil in a greenhouse under the controlled conditions of light temperature and humidity. And then 5 or 7 days later, we collected the grown plants, seedlings, frozen them under the liquid nitrogen, and then made the different analysis of these sprouts. So the question was whether this treated plant acceptor will differ from untreated in phytochemical content and as well in their bioactivity, biological effects, whether there will be room or not. That was the main question. Here is what we have done Firstly, we have using the first phase HPLC method that we have previously developed in our lab and already published. We have used this method to separate, identify and quantify predominant phenolic acids, flavonoids, and L-ascorbic acid or vitamin C in plant acceptor as well as in plant donors, just to see and to quantify the main bioactive components from the group of phenolics and vitamin C in these samples. Here are the results, so the exact numbers. You can see the concentration of vitamin C, different phenolic acids and flavonoids in different types of plant donors. You can bold values are actually significantly higher compared to the other values in the same row. So you can see that the rows has a significantly higher concentration of vitamin C compared to the other 3 plant donors. Then you can see that chamomile has higher concentration of chlorogenic and caffeic acid. It has higher concentration of all the identified phenolic acids as well. Then it has higher concentration of lycopene and luteolin, and definitely apigenin. Apigenin is specific flavonoid for chamomile, so this is something that was completely expected. Then we can see that in Thammus communis there was recorded the highest concentration of camphorol, and the total identified
So I'm sorry, Dr. Ivana, you're just muted. Can you unmute?
So I was muted. Okay. Yeah. So we were interested whether this phytochemical profile will be mirrored in the sprouts of Chinese cabbage that were treated with these plant donors. So we the sprouts that were treated with rose extract will have higher concentration of vitamin C, those treated with chamomile higher concentration of luteolin and apigenin, and so on, those treated with Thammus camphorol. So let's see if this is really so. So here are the results, how the numbers looked in Chinese cabbage sprouts. So this is the control group that was incubated with water only, and treated samples of Chinese cabbage sprouts. As you can see, we have said, okay, the rows had the highest concentration of L-ascorbic acid. However, the Chinese cabbage sprouts treated with this extract did not have the highest concentration of vitamin C, but that was in the case with Thammus communis. This means that this interspecies transfer of metabolites is not just a pure source sink transfer, but some other mechanisms are involved as well. So this vitamin C concentration here is a nice example of that. Then you can also see, if you remember, I said chamomile had the highest concentration of luteolin and apigenin. Here you can see that in the sprouts treated with chamomile extract also had the highest concentration of luteolin and apigenin. So here we can say, okay, the predominant mechanism is definitely source sink phytochemical transfer. It doesn't have to be that it is the only mechanism, but definitely it predominates. Then you can see that camphorol, that was the highest concentration present in Thammus. Here also in sprouts treated with Thammus communis, the highest concentration was present. So here again, the source sink mechanism is obviously the predominant. So you can see that the differences in the concentration of certain bioactive compounds are different, so they are species specific, and you cannot the concentration of a certain compound in a plant acceptor is not predictable without the analysis. So you cannot say if the rose extract has the highest concentration of vitamin C between the plant donors, then the sprouts treated with this extract will also have the highest concentration of vitamin C. It doesn't have to be. So you have to analyze them definitely. So then what we also checked is what is the bioaccessibility of bioactive components in the control and treated Chinese cabbage sprouts. So if you know, so I don't know if you are familiar with that or not, but when you take a water extract or ethanol extract, you take a glass of wine, the bioactive components that you have in a glass will not be the same as in your digestive system, gastrointestinal system, because the extract will, all the bioactive components will react with the matrix of your gastrointestinal system, and the bioaccessibility will be definitely changed. So we looked what happens during the gastrointestinal digestion with bioactive components in sprouts. We have performed this not with patients, not with human, but we did in vitro model of human digestion. So we had 3 phases: initial, gastric, and intestinal. You can see here again, both values are significantly higher compared to the other in the same row. So you can see the concentration of total phenolics in initial, gastric, and intestinal phase. You can see that the sprouts of Chinese cabbage will contain or will show highest bioaccessibility of total phenolics in intestinal phase of digestion in human model after incubation with Thammus communis. Other treatments did not significantly change the bioaccessibility of total phenolics. Chamomile treatment, yes, but only in initial and gastric phase. If we know that the majority of transport of bioactive components to the circulation occurs in intestinal phase of digestion, then we will concentrate definitely on this intestinal phase of digestion. So you can see that total phenolics will be significantly better bioaccessible if you treat the sprouts communis. The same happens with vitamin C. L-ascorbic acid is a form of vitamin C. Again, treatment with Thammus communis will enhance bioaccessibility of vitamin C in all 3 phases of digestion. Ferulic and synapic acid were not, their bioaccessibility was not that significantly changed in the phase. In initial phase, synapic acid, yes, but later on that was in the gastric and the intestinal phase neutralized. Then also quercetin, camphorol, apigenin, flavonoids that we detected in sprouts. We can see that quercetin bioaccessibility will be improved using Hipericum rose extracts and only in initial and gastric or just gastric phase. However, camphorol, which is the predominant flavonoid or flavonol in Chinese cabbage fruits, its bioaccessibility was increased in each phase of the human in vitro gastrointestinal system and with each treatment. So this flavonoid is really susceptible to the treatment with plant donors, and its bioaccessibility is really increased. If we know that camphorol shows a lot of positive biological effects in human organism, this is really interesting result and suggests some further in vitro and definitely in vivo analysis. Apigenin apigenin bioaccessibility was increased after treatment with chamomile in initial and gastric phase. Of course, this was expected because chamomile has apigenin is in the highest concentration among the four plant donors. So this was really interesting. However, in intestinal phase of digestion, apigenin could not be detected at all, which means that it is really susceptible to the digestion in intestinal phase of digestion, and its concentration is really, really low. Then we also checked the antioxidant potential of Chinese cabbage fruits during in vitro gastrointestinal digestion. We used the three standard antioxidant methods, ABTS, BPH, and FRAP. ABTS is known as the most sensitive, and with this method, you can see that we recorded the significantly increased antioxidant potential of Chinese cabbage fruits after treatment with Hypericum, rose, and Thammus communis extracts, and this was improved in intestinal phase of digestion. So it is really interesting. So we can really improve the antioxidant potential of these plant material by using this interspecies transfer of metabolites. Then we tested what happens with the antidiabetic potential of Chinese cabbage fruits before and after. So after treatment with each of the plant donors, and of course in comparison to the control plant treated just with water. So if we know that diabetes 2 is a disease related to the activity of amylase and glucosidase, two major enzymes responsible for increase of concentration of glucose in the blood. If we can inhibit these enzymes, probably we can actually decrease the concentration of glucose in the blood and actually help the people fighting diabetes. So we decided to check the activity of extracts of Chinese cabbage fruits, control and treated, against alpha amylase enzyme. If we can increase the inhibition of this enzyme, we can definitely perhaps help to the fight against the diabetes. So on this vertical axis, you can see the percentage of inhibition of this enzyme, and if you can just see here, you can see the inhibition percentage of control plants, and then you can see that after treatment with Thammus communis, the inhibition of this enzyme was significantly increased, which is really interesting. It shows the potential of Thammus communis treatment of Brassica rapa-pekinensis or Chinese cabbage sprouts. Definitely suggests further in vitro investigations. Then we also looked at the anti-proliferative effect of Chinese cabbage fruits. So we have tested the effect of extracts of Chinese cabbage fruits on the proliferation of cancer cells, MCF7, those are human breast cancer cells, and human embryonic kidney cells as a type of healthy cells. So one always wishes for itself to find the extract that will act anti-proliferatively against cancer cells, but will not stop the division of normal healthy cells. And this was accomplished by Chinese cabbage fruits treated with Hypericum, rose, and chamomile at the concentration of extract 100 milligrams per kilogram of fresh weight. You can see that these treatments decrease the proliferation of human breast cancer cells, however, did not change the proliferation of healthy human embryonic kidney cells, which shows the potential of this phenomenon in the fight against the proliferation cancer cells. Here with Hypericum perforatum, given concentration of 25 milligram per kilogram, acted against the proliferation of breast cancer cells. However, healthy cells did not, their proliferation was not was not changed. So definitely 125 milligram per kilogram of extract of brassica or Chinese cabbage fruits treated with Hypericum perforatum could be further analyzed, as well as these two samples at the concentration of 100 milligram per kilogram definitely suggest further in vitro investigations. We also checked whether the treatment with extracts of plant donors could actually change the expression of genes in plant acceptor. We have concentrated on the genes in phenylpropanoid biosynthetic pathways, namely phenolics, how synthase, it's the first gene in this phenolic biosynthetic pathway, and flavonol synthase. It's an enzyme that is responsible for synthesis of the dominant flavonoids in Chinese cabbage fruits. Those are kaempferol and quercetin. Here you can see what happens. We have measured the expression at 2 time points, 4 hours after incubation of fruits with plant donor extracts and 5 days. You can see that the expression of gene is time specific and also species specific. First, what you can here see is that during the incubation or treatment with Hypericum perforatum extract, the expression of genes Halcon synthase and flavonol synthase was reduced in Chinese cabbage fruits, and Halcon synthase was far more reduced than flavonol synthase. This is probably because Halcon synthase is the first enzyme in biosynthetic pathway, and if you decrease its expression, then later on the expression of flavonol synthase will also be decreased, because the precursors for flavonol synthase will be present in lower concentration. You can see that the level of gene expression was more reduced after a shorter period of time of incubation of Chinese cabbage fruits than after longer period of time. Here with Rose, the results were opposite. So a higher decrease in gene expression was recorded 5 days after incubation than 4 hours. And here, 4 hours after incubation, you can see that flavonol synthase expression was even increased compared to the Halcon synthase, whose expression was decreased. So we can definitely see that there is an effect of plant interspecies transport of plant bioactive components definitely affects the gene expression in plant acceptor, and this is gene specific, time specific, and definitely species donor specific. So this phenomenon of interspecies transfer of metabolites definitely has an effect not just on phytochemical profile of plant acceptor, not just on its activity, but also at the expression of genes in this plant donor. So the interspecies transfer of metabolites is really deep consequences on the plant acceptor. After we gathered all these data, we have processed them in a statistical way. So we use principal component analysis, a method to present all the results in a simplified way. So in this statistical method, you actually all the data you make you present as two principal components. You can see based on the variables that you have measured. Here are those different plant bioactive components. You can see how plant donors are actually organized. You can see that Thamus and Hypericum are more similar to each other based on the set variables than Rose or Chamomile. Then also made this with the control and the treated Chinese cabbage fruits. You can see that control fruits are here. They differ significantly in their phytochemical profile from the treated groups. You can see that Chinese cabbage fruits treated with Thamus and Hypericum are more similar to each other, while those treated with Rose and Chamomile are also similar to each other, but both of these differ significantly from the control group based on the phenolic compounds identified in the analysis. And also you can see which of the phenolic compounds mostly contributed to the Chinese cabbage sprouts in each group. So you can see that these group of Chinese cabbage sprouts treated with chamomile and rose, they have higher concentration of alpha-genin, caffeic acid, luteolin. You can see that those treated with Thymus and Hypericum, higher concentration of lecithin, camphorol, and total identified flavonoids, phenolics compounds. So very a nice way to gather all the data together and make it more easy to visualize. We made the same analysis with the samples during the initial gastric and intestinal phase of digestion. You can see how the samples grouped at the initial phase, how they grouped in gastric phase of digestion, and how they grouped after intestinal phase of digestion. You can see that the grouping is changing, and that was expected. Namely, when you take in your plant food, their bioactive components will interact with the matrix in your gastric system, in your intestinal system, and definitely will change. So the phytochemical profile and bioactivity of a certain plant food definitely depends on the matrix that you have in your own gastrointestinal system, and here is how they differ. So you can see that intestinal phase of digestion, which is important because the highest level of absorption of components into the circulation occurs here in this part of digestion. You can see that Chinese cabbage sprouts treated with rose are most similar to the untreated components, while those treated with Hypericum or Thymus differ most significantly. So the treatment with Thymus and Hypericum made the biggest change in the phytochemical profile and bioactivity of Chinese cabbage sprouts. Based on all these results, we can definitely, so this is the first assessment of nutritional significance of interspecies phytochemical transfer. We can say that Thymus communis, you can see camphorol and total phenolic contents in Chinese cabbage sprouts. Thymus communis also improved antioxidant and lipoglycemic activity of sprouts, while the antiproliferative activity against human breast cancer cells of Chinese cabbage sprouts was improved by the treatment with Hypericum perforatum, rose, and chamomile. And definitely the transfer of phenolics and vitamin C to acceptor plants was affected by donor matrix or plant donor species. And at the end, we can say that interspecies transfer of metabolites is definitely a promising approach to improve sprouts nutritional properties and improve can actually offer some new food on the market. So definitely we can answer the question from the beginning of the presentation. Treated versus untreated Chinese cabbage sprouts, definitely treated are improved. Their phytochemical profile, their antioxidant activity, their antidiabetic potential, their antiproliferative effect against human breast cancer cells. So this between species, transfer of bioactive compounds between different species is definitely a bright future. All these results we have published last year in the Food Research International. In this paper you can find more details about this, what I have now introduced to you. If you are interested in something more, definitely you can find there, or just send email and we can discuss it further. Here are all the collaborators on this work. So this is the group I'm working in. We have done all the phytochemical analysis, anti-diabetic tests, and statistical analysis. Maria from Bošković Institute, she was responsible for anti-proliferative testing, and the group from Germany was responsible for qRT-PCR analysis of gene expression. And before I end my presentation, I'd just like to say two things more. Currently, I'm a special issue editor in the journal Plants. It's a Q1 journal with impact factor 2.762. It's covered in PubMed, current contents of all the relevant databases. The issue I'm working in is called Viability plant specialized metabolism, causes, consequences, and possibilities. If you have any unpublished data so far that could fit, you can send us the manuscripts. The deadline for submissions 31st January the next year, so there is plenty of time. More details you can find under this link. I can assure you that I will give your manuscripts to the appropriate reviewers in 1 week time, and they will have the next 2 weeks to make the decision. So in altogether in 3 weeks you will get the decision. You will not have to wait for a long period of time. So if the results are interesting, then they will be published in the journal. So please, if you have unpublished data, just let me know. And finally, at the end, I also would like to present this project. So this year, I managed to get the project with the title Indirect Effect of Global Warming on Mammals' Physiological Parameters by a High Temperature Stress Plant Diet, namely in collaboration with animal physiologists, veterinarians, mathematicians that will do the models. We will investigate the effect of global warming on plant model, seedlings and grown mature plants. The model will be broccoli. Then later on, these plants that will be significantly changed, they will be fed to the mice as mammal models, and we will look into the different physiological parameters of mice, that just to see whether they will be changed accordingly to the change of phytochemical profile of plants under high temperature stress. The resources that we gained were from Croatian Science Foundation. It's a bit more than 180,000 euros. We have a period of 4 years ahead. We have started officially in February. The plants have the first biological replica of plants is already grown at the seedling stage, and tomorrow we will start the first analysis. If some of you would be interested for collaboration on this theme, just let me know, send email, I will be glad to discuss it with you. And I think this is all. So thank you once more for your appreciation. Thank you.
Yeah, thank you very much, Dr. Ivana Sara, for your nice presentation. So in fact, it's something different and very
You're
interesting. Yeah, in fact, it's something different and very interesting that the interspecies transferring of metabolities may enhance the nutritional properties. So it's a very nice presentation, and I would like to go for the participants now. Do you have any queries to discuss with Ivana Sara? You're most welcome now.
Yeah, Ivana, I'm Kakuli Banerjee again from India. I belong to the Department of Biodiversity and Conservation of Natural Resources. A very simple question is, one of my scholars is working on the same aspect. So I just wanted to have a clarification on how the plant phytochemicals and antioxidants act on the human body. How they are helpful in the human body in stress relieving of free oxygen or free radicals in the human body. If you have any idea on that, if you can throw some light on that. Thank you.
I haven't heard you very well because can you repeat the question? I didn't hear you at all.
I just wanted to know, because in plants you have shown that there are many phytochemicals and antioxidants on which you have worked. For example, vitamin C and vitamin E, all these properties that we have in the plants. Now, how that can be utilized in human body, in solving the problems in human body?
So your question is, can we utilize this vitamin C from the plants in our body? That is the question.
Yes. How can we use those plants for our own human body?
So first of all, vitamin C, the most well-known activity of vitamin C is as an antioxidant. So it's in our body. It will act as an antioxidant. It will fight the free radicals in our body. So you can just improve the concentration of vitamin C in your body.
But I mean, can it be directly used, or we can use it in some product form? Because in some plants are not edible. The plants which are not edible,
of course.
Yeah.
Well, of course, if the plant is not edible, if it is toxic, then you won't take it.
Yeah.
So you will just you will take a precaution. You will just take plant donors, those that are not toxic, because toxic compounds can also be transferred. That's the point. Toxic compounds can also be like nicotine, okay? But then you have to be very careful and always choose the plant donors that you know that are healthy. So Thammus communis, for example, you must use just young shoots, because if you take the mature plant, that plant in mature stage has a lot of toxins, and it wouldn't be advisable to take it.
Right.
So that's something that you have to be aware of. So always, for sake, just use the plants that you are safe, that you know that are safe, and that they do not have toxic compounds. So toxic compounds can also be transferred, and that's something that you don't want.
Yeah. Yeah. Thank you. Thank you. Thank you.
Welcome.
Thank you very much once again, Dr. Kakali Banerjee, for your queries.
Bye.
And thank you, Dr. Ivana.
You're welcome.
Yeah. Yes, now I would like to move on to the next presenter. So we have some poster presentations now. So I would like to invite Joanne Ko from South Korea. Dr. Ko is an ecological agronomy researcher serving as a professor at Jeonnam National University, Gwangju, South Korea. His research focuses on agriculture remote sensing and crop modeling to develop and deliver efficient crop growth and productivity monitoring practices for various aspects and scenarios and different geographical regions. Dr. Ko, you're most welcome to begin your presentation now. Thank you.
Ask to unmute.
Dr. Ko, can you hear us? Can you unmute your microphone, please?
I'm sorry, my microphone was off, so I'm gonna show my face and then let you guys share.
Yes. Yes, we can see you. Thank you. No worries.
So,
no worries.
Okay. And then I'll share my screen first.
Yes.
And then load the poster I prepared.
Okay, no worries. Take your time.
All right. Nice to meet you, ladies and gentlemen, and thank you for attending the conference. And what I'm gonna talk about is integration of machine learning and deep neural network, so-called deep learning approaches, into a process-based crop model for prediction of crop growth. In this study, we think that deep neural network or deep learning and machine learning techniques can be promising tools to advance a mathematical crop modeling methodology based on integrating these schemes into a process crop model to predict crop growth, especially for prediction of crop growth. So in this study, we intend to propose development of an innovative hybrid approach of crop model and integrating remote sensing data. So our objective here is a little small. I'm gonna make it a little big. Today I present an effort to integrate ML and DL methodologies to a process-based mathematical crop model. We investigated suitable DNN or DL models for the weather data prediction to estimate the leaf area index of rice based on the relationship between leaf area index and weather factors using ML regression models. I'm sorry. So in this study, the study location of interest was an experimental field in Choron, and the whole geographic regions of the Choron and Paju counties are here on the border of South Korea and North Korea. And we used rice growth and yield data from this study site in Choron, experimental site, from 2011 to 2015. Also, we simulated crop growth and yield using a mathematical crop model that uses remote sensing data in order to make big data or to combine a model that uses remote sensing data with DL or ML model. Machine learning and DNN models investigated include 9 regressors included in scikit-learn and 1 DCNN and LSTM, RNN, 4 DNN in Keras. Actually, this one is a still ongoing process. So I didn't include this output in this presentation. Sorry about that. But to simulate to reproduce crop growth and yield, we used a model, a mathematical model that can simulate leaf area index and other growth factors together with crop yield, assimilating remote sensing data. Here is the part that machine learning and or deep learning model can be integrated in this part. Okay, to instead of using remote sense data assimilated here, I'm intending to make machine learning or deep learning algorithms integrated here. And then here is some result, preliminary result I pulled out and I have for now. As I presented just before, say 9 regressors, machine learning models used, and test scores show the whole 9 regressors ranged, the score ranged from 0.76 to 0.89. And then I'm showing here the result plotted, the scatter plot between the relationship between simulated leaf area index using regressors actually observed leaf area index. The panel A shows the training for this one, and panel B shows test scores, test result. This is for the output in this Choron district region, whole the region, and the other one is for the result for this administrative region, which is Paju. Okay, I used 2 geographical regions, and this output result is pretty similar, but slightly lower than this region, but it ranged from about 0.70 to 0.89. And then I also plotted the best train output with the scatter plot between a simulated leaf area index using random forest and then observed leaf area index. And then this one shows the test output score. And using this random forest regressor, what we can do is the already developed model that uses remote sensing data, and then we can predict or simulate leaf area index, and then use as an input to simulate and crop growth. And this is the result of simulated leaf area index using the model that uses remote sensing data. And then, as you can see here, simulated leaf area index matches well with observed leaf area index, reproducing yield in close agreement with observed yield. And then lastly, what we are trying to is using these predicted leaf area index using machine learning or deep neural network, which will be as ongoing research, and then we are working on. We will reproduce crop yield and growth for the whole the geographical regions of study areas of interest, which is Choron and Paju in this case. So here is my study summary. The Quorum study has investigated the modeling performances of available machine learning regression models to simulate leaf area index using climate data, determining the best working model of random forest for rice, as I showed just before. And then the whole 10 model machine learning regression models ranged from 0.676 to 0.90 for Choron, and from 0.70 to 0.89 for Paju. So we assume that equivalent performances are likely achievable using deep neural network models in applying big data, which I didn't include it here. I'm still working on. I was thinking to include this part, but I was a little behind. Again, I'm sorry about that. So we'll further report our ongoing effort and result to simulate crop yield later. Thank you. That's about it. It was a little short, but that's it. Thank you for your listening, and I'll get your questions if you have.
Yeah, thank you very much, Dr. Ko, for your nice presentation.
Thank you.
So do you have any queries to discuss with Joanne Ko?
Yes,
Yes, please.
I have a question.
Yes.
Okay, so I'm not sure if I have understand everything, but it definitely drew my attention, and I was wondering, would you be interested in collaboration perhaps with us, because we are not so good in prediction models, but this is something that we are definitely interested in, and for the next project we have made already a team with mathematicians that do statistics, but people like you could also help us. So would you be interested in possible discussion, for example, tomorrow, just so that you can say to us what exactly you can do, and perhaps we could do something together?
Sure. What crop are you interested in simulate or predict the variable? What
Yes.
crop are you interested in, and what variable are you interested in simulate?
I didn't hear you. What?
What kind of crop are you interested in working with? The crop species. Crop, plant. What kind of plant? What kind of crop?
What crop species? What species? So
crop?
Currently, I didn't understand the word crop. Okay, currently we are working on brassica, Chinese cabbage.
Chinese cabbage.
And broccoli.
Broccoli. Oh, man, that's
yeah, Chinese cabbage and broccoli.
A little challenge, I think.
These are brassicas.
I'm mostly working with agronomic crops, food crops like rice, wheat, barley, corn, things like that. So brassica is slightly out of my interest area. Yes.
So, okay. Well, I just wanted to ask. I need to ask. Okay, thank you.
Yeah, thank you.
Okay, of course. Always. Thank you.
No problem, yeah.
Study, any other queries for Dr. Ko?
Are you interested, Professor Kaul, in forest plants?
Forest plants.
Forest, I mean trees, especially trees.
Uh,
Or you're
I
strictly on the food crops only?
I
Because I asked is because nowadays a lot of work on forest leaf area index and its prediction model using machine learning is going on. So
yeah.
since India has a very good patch of forest, both terrestrial and as well as mangroves. So I was just interested to ask you once. I was inspired by Dr. Ivana because once she asked that, so I thought, why not? I should also put forward this proposal to you.
Thank you for your interest.
Of course.
In my whole career, have been working only
On rice.
with agronomic crops.
Okay.
Whole different agronomic crops: wheat, corn, soybean,
As such,
things
sir,
like this.
if
So yeah.
if that, yeah, if that is the case, I mean, what type of data is actually needed for rice? Because the place where I stay, it is said to be the origin of rice in India.
Yeah,
So,
I know.
yeah, Koraput is a place in India which is known for its origin, and Professor Swaminathan is one of the scientists in India you know very well who is working on rice and is a legendary figure in rice scientist. So, I would like to know what type of data you actually need to predict in terms of your machine learning and prediction model for the production of rice in India or any type of stress tolerance. What type of data is required for that?
Actually, the reason I have been working with this uh, prediction of leaf area index is I was preparing a big project. I fund it now to monitor East Asian rice growing areas, which include North and South Korea, Japan, part of those eastern part of China. We may include, as you said, India. What I'm now collecting is satellite imageries of the research interest, and then maybe if we have the experimental location data from experimental location of interest, which can represent the region of our interest. If we can include India, maybe that we can work with, and then the variable our interest is without doubt grain yield. And then and another one is the leaf area index. The reason why I'm interested in the leaf area index, we can make a relationship between the crop condition from remote sense data with leaf area index. That's why I'm sticking with leaf area index.
Mm-hmm.
So
no, I mean, for ground data, what type of data you are using for ground data?
leaf area index.
Only leaf area index.
And then the climate
Grain
data.
production.
Grain production, of course.
Yeah.
And then climate data, solar radiation, temperatures, low and
Yes.
high temperatures. And that's a minimum requirement, actually.
Yeah.
And some, crop management record will be helpful to simulate crop conditions. Yeah. Yes.
So a coordinator can help us in coordinating if required.
What do you say?
I told our coordinator for the conference can help us in if at all it's required.
Yeah.
Yeah,
Thank
sure,
you.
Dr. Kakoli Banerjee.
Yeah.
We'll do that. No worries.
Yeah. Thank you. Thank you so much.
Thank you. Thank you very much.
Thank you very much for your questions and your nice response to the participants, Dr. Kill. And I would like to thank you once again for your presentation.
Thank you very much
You're
for your,
welcome. Yeah, you're
time,
welcome.
you know.
You're welcome.
Okay. Bye-bye.
Yeah. So, and I would like to go for the next poster presenter, Ms. Erin Debussy. Ms. Erin studied biology in University of Athens, from which she graduated in 2017. Since then, she is a PhD candidate in molecular plant physiology in the same department, funded by the State Scholarships Foundation, and has published 2 papers. Ms. Erin, you're most welcome to begin your presentation now.
Good morning from Greece.
A very good morning.
Can you see the presentation, the poster?
Yes, we can say perfect. Please go ahead.
Perfect. Thank you for the introduction and for giving me the opportunity to participate in this webinar. In our lab, we're investigating genes that their products interact with the selenium-binding protein, SBP1. And in this study, we focused in a papain-like cysteine protease, the RD19C, and its ability to interact with SBP1. Firstly, it is important to explain why we are studying SBP1. Generally, selenium-binding proteins are very interesting as they are highly conserved and have representatives from all life kingdoms, including archaea, even humans. As its name indicates, SBPs have the ability to bind selenium in a covalent manner, creating a new protein family different from the selenoproteins, which includes modified selenocysteine amino acids. Furthermore, SBPs have many conserved metal-binding motifs with the ability to also bind cadmium. In humans, there are plenty of studies that they have shown its correlation to cancer and other diseases. Moreover, it has been proposed as a pharmacological target as well as a biomarker to schizophrenia. Recent studies have revealed that the bacterial methanothiol oxidase belongs to the selenium-binding protein family, and it was demonstrated for the first time that the human SBP has also the same activity. In Arabidopsis, the model plant our studies are focused on, there are 3 alleles and they participate in responses to selenium and cadmium stress. SBP1 has been considered as a general response gene by participating in a protein network as it has the ability to interact with proteins related to protein trafficking, membrane biosynthesis, and redox cell control. As it was previously mentioned, in this project we investigated a papain-like cysteine protease, the RD19C. Proteases hydrolyze peptide bonds participating in a proteolysis procedures. Proteases that belong to this family have been proposed to lead to program cell death under metal stress. There is no more much information about RD19C. However, it is known that it is located in vacuoles and participates in anther development by mediating programmed cell death. Moreover, omic studies have showed its induction in response to cold, drought, and nutrient deficiencies. Now, the aim of this study was the investigation of the RD19C and its interaction with SBP1 by using classic molecular techniques such as gas staining, quantified qPCR, protoplast isolation and transfection, and yeast to hybrid assays. To investigate the implantar interaction, bimolecular fluorescent complementation was used in a protoplast system in which a fluorescent protein is split into 2 fragments, each one fused to our genes of interest. Only if these two proteins interact, the fluorescent protein reforms, being able to give us a signal. In order to investigate the transcriptional activity of RD19 promoter, we generated stable plants containing a 428 base pair sequence upstream the open reading frame, fused to the gas reporter gene. Gas expression was detected in hadothodes, stomata, leaf hairs, as well as the vasculature of cotyledons and the first emerging leaves. Furthermore, it was expressed in the vascular cylinder of the main root, as well as the lateral roots. The expression pattern of SBP1 has been described before, and the reporter patterns are overlapping with those of RD19C, indicating that the transcripts of these genes colocalize. To study the protease response to chemical treatment, we initially studied the promoter activity after treatments with selenium and cadmium. The major expression change was observed in selenite-treated plants, where gas signal was extended in the root tip of the main root, and in the other two compounds, there were no observed differences in the expression pattern. However, the staining in selenate was weaker, whereas in cadmium were more intense. Additionally, the relative expressions of RD19C mRNA was studied in seedling roots after the same treatments. In this analysis, there was a twofold upregulation in the presence of selenite, a significant downregulation in selenate, while no notable change was observed in cadmium. This is in contrast with SBP1 expression under the same treatments that exhibited a reverse response in selenium compounds and a remarkable upregulation in cadmium. These experiments indicate the differential response of RD19 promoter, at least in selenite. In order to clarify the physical ability of the protease to interact with SBP1, it was performed a yeast-2 hybrid assay. In this experiment was tested all three alleles of SBP, and it was confirmed a strong positive interaction only with SBP1. To investigate the subcellular localization of genes of interest and confirm the implantam interaction of them, Mesophil protoplasts were isolated and transfected with the appropriate plasmid constructs. RD19C is targeted in a globular structure near the nucleus, appearing as a small vacuole, distinct from the main central vacuole. SBP1 was detected in nucleus and cytosol, whereas the interaction was captured only in cytosol. In conclusion, we saw the colocalization of the RD19C and SBP1 transcripts in hadothodes, stomata, roots, and lateral roots. It is known that hadothodes are the primary sites of auxin biosynthesis, and there are studies which show that both genes are induced by this phytohormone. Moreover, selenium compounds affect the relative mRNA expression of RD19C and SBP1 in a reverse manner. Papen-like cysteine proteases lead to programmed cell death under metal stress. Thus, we can assume that RD19C has the same role in response to selenium. Furthermore, we examined the subcellular localization of both proteins, with the protease targeted to the vesicle network and SBP1 to nucleus and cytosol. Investigating the protein interaction between these proteins, we showed that it takes place in the cytosol. We suggest that RD19C is part of the SBP1 protein network, which could participate in programmed cell death under the conditions that interaction takes place, probably following selenium stress. However, further investigation is needed in order to understand the precise conditions under which the examined interaction occurs. Thank you very much for your attention and for your time.
Yeah, thank you, Irene. Does anybody has a question for Irene and her presentation? If not, then I would like to thank you for your poster presentation. Keep my fingers crossed for your PhD.
Thank you very much.
And more papers on this theme.
Thank you.
I know how it is. You're welcome. Yeah, we can go on. We can go further.
Yeah, thank you very much, Irene and Ivana, for your discussions. So yes, we'll move on for the next presentation. Yeah, hopefully Dr. Alam. Dr. Alam worked as lecturer of pharmacognosy and natural products since 2017 at the Faculty of Pharmacy, Ministry of University from Egypt. She has been teaching pharmacognosy for undergraduate students, and her research work is in the field of natural products isolation and structural elucidation. Also, her part of work was in plant tissue culture and natural products analysis using HPLC. She's currently interested in screening for bioactive secondary metabolites for cancer treatment and antibiotic resistant bacteria. Dr. Alam, you're most welcome to begin your presentation now. Thank you so much.
Okay, thank you. Our talk about metabolic profiling and anti-cancer and anti-inflammatory screening for some wild plants growing in Egypt. This work done by Dr. Dalia Al-Amir Muhammad and Dr. Muhammad Ahmed Zaki and Dr. Marwa Hassan Al-Saif, and well presented by me, Ahlam Hashim. The desert area in Egypt comprises about 96% of its geographical area. Many wild plants growing in this desert. 3 wild plants were collected and subjected for our work. These plants are Fagonia critica, Volitaria libia, and Trigonella stellata. First, talk about Fagonia. Fagonia is a genus belonging to family Zygophallaceae and comprises about 18 species in Egypt. Phytochemical screening about this plant showed that the plant containing saponins, triterpenes, flavonoids were detected in this plant. Recent researches about traditional uses of Fagonia indicated its use for treatment of fever, typhoid, liver disorders, asthma, and tumors. And also it is used for some digestive disorders and cancer. And also it is used in some skin disorders as boils and eruptions. Also this effect is reported for Fagonia. Pharmacological study about Fagonia reported to as antimicrobial and anti-inflammatory and anti-cancer activity, and especially used for Salmonella typhi infection. Voltaria is a plant belonging to family Composta, and it is growing wild in Egypt and belonging to the genus Voltaria, which contains about 18 species. Previous phytochemical studies on Voltaria reported that the plant containing sesquiterpene and flavonoids. Flavonoids as nactofluorine, isofloxacin, and isoquercetin were isolated from the plants. Antioxidant and antimicrobial and anticholinesterase and antidiabetic activity of this plant were evaluated. While Trigonella stellata, which belonging to family Legumineaceae, Trigonella also containing Trigonella pneumogrecum, which is used traditionally as antidiabetic and antihyperlipidemic. Many phytochemicals were detected in this genus as phenolics and isoflavonoids. Previous study about Trigonella stellata reported the isolation of new isoflavonoid compounds and antidiabetic activity of the alcoholic extract of the plant and the isolated compound also evaluated. Aim of our work: detect phenolic and flavonoid content in the target plant, detect different metabolites in the extract of 3 plants using UPLC mass techniques, also evaluation for in vitro cytotoxicity and anti-inflammatory activity of these plants. Metabolic procedure: 1 gram of the ethanolic extract was subjected to metabolic analysis using analytical techniques. The mobile phase composed of purified water and acetonitrile, which containing 0.1% formic acid in each solvent. The total phenolic content was determined using Folin-Sciatic reagent, which is used for measurement of total phenolic. The procedure is as follows: 300 microliter of the methanolic extract was mixed with 0.2-0.5 milliliters of Folin-Sciatic reagent, then 0.2-0.25 milliliters of sodium carbonate was added to the mixture. This mixture stands for 19 minutes at room temperature. Then spectrophotometric was used to measure absorbance at 700 to 5 nanometers. Gallic acid was used to prepare the standard curve, and we calculate our result as milligram of gallic acid equivalent in 1 gram of dried plant sample. While total flavonoid content was determined as using aluminum chloride reagent and as follows: 0.5 milliliters of the methanolic extract was mixed with 0.2-0.5 milliliters of distilled water. Then 0.15 ml of sodium nitrate solution was added to the mixture, and 0.3 milliliters of 10% aluminum chloride reagent was added to the mixture, and UV absorbance of the mixture was measured at 510 nanometers. Our result is calculated as microgram rotten equivalent in 1 milligram of the dried plant sample. Antioxidant assay. Antioxidant assay of the 3 plants was determined using DBPH as free radical model, and the procedure as follows: 300 microliter of the sample was mixed with 3 milliliters of DBPH solution. The mixture stands for 30 minutes in the dark in the room temperature. Then spectrophotometric used to measure the absorbance at 517 nanometer, and using ascorbic acid as positive control. Cytotoxic activity of the plants was determined as using MTT assay and against 3 cell line, human liver carcinoma, human breast carcinoma, and human colon carcinoma. Dose response curve was generated, and IC50 value were defined as the concentration of extract required to inhibit the cell proliferation by 50% concentration was calculated. 5-fluorouracil was used as positive control. While the anti-inflammatory activity of the plants were determined by COX inhibitor activity, COX-1 and COX-2, and nitric oxide inhibitory assay using Elgerez nitrate assay. Our result, LC mass dereplication result of Fagonia critica ethanolic extract showed that 15 compound was detected in Fagonia. These compounds mainly fatty acids, flavonoid glycosides, sterols, butyrocarbons, ditropine, and this is the compound detected in Fagonia critica extract. While results of Trigonella stellata showed about 20 compounds, these compounds are classified as stilbene, phenolic acids, flavonoids, steroidal saponins, amino acids, isoflavonoids, butyrocarbons. While LC mass result of Volateria Libya ethanolic extract led to the replication of 28 compounds. All compounds detected in Volateria are sesquiterpene compounds and flavonoids. Results of cytotoxic activity showed that Fagonia critica extract is the most active as cytotoxic compound against 3 tested cell lines. While anti-inflammatory activity showed that Volateria Libya is the major active compound or major active extract as anti-inflammatory, especially against COX-1 and COX-2. Concerning the total phenolic flavonoid content and the antioxidant activity of the 3 extracts, we found that Trigonella stellata is the plant containing a high content of phenolic compound and flavonoid, while Volateria Libya is the major plant concerning antioxidant activity. Conclusion: metabolomic profiling led to identification of 15 compounds from Fagonia critica, 20 compounds from Trigonella stellata, and 28 compounds from Volateria Libya. The compounds detected in Fagonia were belonging to fatty acids, triterpenes, sterols, flavonoids, butyrocarbons, and ditropine. While the compounds detected in Trigonella stellata were stilbene, flavonoids, amino acids, phenolic acids, steroidal saponin, ditropine, and isoflavonoids. While compounds detected in Volateria were sesquiterpene and flavonoids. Cytotoxic activity of 3 extracts revealed that Fagonia was the most potent extract as anti-cancer, while Volateria is the most potent extract as anti-inflammatory. Thanks.
So thank you for your nice presentation. I find it really interesting because it is quite similar to what we do in our lab. So I have a question. Our first question is, are these results published? Have you published these results already? Ahlam, do you hear me?
Okay, تفضل. Okay. Any question?
So have you published your results? Yes, can you publish your results?
No, still not published yet.
Are you planning to?
Yes, we plan to publish it.
I didn't hear you.
Yeah, we
I think
plan
our
to
internet
publish.
connection is not working.
We plan to publish it.
Because I would suggest, just to add statistical analysis, because you have nice numbers, nice exact data, but no statistics. Can you do the statistics? Can you hear me, Ahlam? Yeah, we lost the contact. I think Ahlam doesn't hear me.
Yeah, it's maybe it's net due to net poor connection. We lost her connection. No worries.
Yeah. Okay, I will write to her in chat or something.
Yeah, that would be great.
Yeah.
Yeah, okay,
Yeah,
Dr.
she's not here anymore. Okay.
Yeah, okay, Dr. Ivana, we will move on to the next presentation. So, and here is our last poster presenter, Mr. Yusuf Mohammed. Is a student at fourth year from Faculty of Agriculture Biotechnology Department at Al Azhar University in Cairo. He holds training in basic protein isolation and purification from City of Scientific Research and Technological Applications, training at the Desert Research Center in Tissue Culture Technology, and applications of bacteriology, virology, immunology, and serology in National Research Center, the applications of QC and QA in pharmaceutical industry at Jawad City of Science and Technology, applications of pharmaceutical industry in Memphis for pharmaceutical and chemical industries. His basics of digital marketing from Google, IB Europe, and the Open University, and he attended the Global Entrepreneurship Week at American Corner, and in the final stage to publish his first research in a scientific journal. Mr. Yusuf Mohammed, you're most welcome to begin your presentation now.
Okay, thank you. I will share my presentation.
Yes, you can share your presentation now.
Okay. And now we can see.
Yes, we can see it.
Yes. Thanks so much. Hello, everyone. First, I would like to thank the NAT Research Forum for inviting me to this conference. Today, I'm delighted to present part of my research work and talking about plant biotechnology and synthetic biology because it's a great importance in our lives. All living organisms have the ability to improve themselves through natural means in order to adapt to changing environmental conditions. However, it takes hundreds of years before getting any improvement. Then humans learned how to domesticate and breed plants in order to develop crops to provide his needs. This be by using biotechnology. Biotechnology is defined as a set of tools that use the living organism or a part of it to make or modify products and improve plant, trees, or animals, or even develop microorganisms for specific use. It includes a number of tools as bioinformatics, microbiology, molecular genetics, biochemistry, plant physiology, and microbiology. With these items, I am going to talk about are first, I will talk about the history of plant biotechnology, then I will talk about plant biotechnology and why plant biotechnology, then impact of plant biotechnology and medical plant with biotechnology and synthetic biology. Last thing, I will talk about my work in the lab. History of plant biotechnology, represented in two types of breeding. First type is classical plant breeding, represented in founding of genetics science and crossbreeding to synthetic trait, that I mean crossing over. And another type is modern plant breeding, represented in mutation breeding and green revolution in 1960 to 1970, and plant tissue culture breeding. From 10,000 years ago to this day, plant technology evolved from simple selection to genome editing. As we see, simple selection, then controlled crosses, then hybrid crosses, then markers, then genetic engineering to genome editing. The science of genetic began when George Mendel discovered the law of inheritance by studying flowers in his garden. We can say that agriculture biotechnology is genetically modifying crop science. While we know that there has been different forms of agriculture technology development for more than 10,000 years or so. Genetically modified crops or transgenic crops produced by transferring genes where DNA transfer could be from other crops or even from bacteria or virus. Finally, it can be said that plant biotechnology is found on the role of circular tissue potency and genetic transformation. As example, in 1996, there was the first large-scale cultivation for genetic modified soybean and genetic modified maize. Now, I have a question. The question is, what is the difference between traditional breeding and GMO or genetic modified organism? They are similar in altering crop genes with artificial selection 4,000 years ago, and there is no fundamental difference. And they are different in genetic engineering can mix genes of very different species, and genetic engineering is in vitro lab work, not with whole organism. Genetic engineering use novel genes. And another question is why we use plant biotechnology, or why we use biotechnology in plant. Plant biotechnology continues to evolve with new techniques that will advance food production for farmers and meet the need of customers. These techniques as plant culture technology and gene transferring. Now let's talk about biotechnology and its role in enhancing to the benefit of this plant with massive technological progress that the world is going through. Biotechnology has used in the analysis of plant genetic makeup and studies the chemical compounds and studies the effect of these therapeutic compounds and or to multiply this plant in vitro to produce these medical compounds in vitro too. A plant micropropagation or tissue culture involves the use of small pieces from plant tissue that it called explant, which we are cultured in feeding medium under sterile condition. By using different concentration from growth regulators for each explant, plant can be induced to produce new shoots and the production of secondary compounds too. Now, I am going to talk about the impact of biotechnology and what we get back from using it. First, the economic impact. With the full use of biotechnology, overall income for all regions measured by gross national product is estimated to rise by $210 billion a year. The environmental impact represented in pesticide reduction and avoid the damage that cause to the environment. As health impacts represented in more food and better food and more healthy food. When we talk about society impact, with biotechnology, we could help address many global problems, such as climate change, aging society, and food security, and energy security, and other problems. And the introduction of many products that serve all areas as therapeutic products, as antibodies, vaccines, growth stimulators, proteins, and bioplastics as polyesters like BHA or BHB and other products. When I talk about medicinal plant, we found medicinal plants are very important in our life due to what this plant contain of compounds used compounds used in the treatment a lot of diseases since ancient times. People have used this plant in his treatment, and this plant indispensable whatever the world developed around us. So we must consider in using this plant and making full use of them because of their positive impact for human health. Worldwide, more than 50,000 plant species are used in medicinal purposes because its plant are rich resource of bioactive compounds. Moreover, plant extracts have been used to prevent and to treat a lot of diseases. Finally, there is a global demand for green production process to be available in a timely. When we talk about synthetic biology is fast growing and have a lot field as chemistry, physics, engineering, and mathematics. It's defined as the design and develop a new artificial biological compounds for complex biological organisms, such as enzymes and cells, or design of biological system for novel application. This emerging field should play an important role in future agriculture for improve the traditional crops. This improvement as phytochemicals that we use it in the treatment for more healthy life. The last thing I will talk about it, my research in the lab. I choose the vinca plant because it's one of very important plants because it's contain very important alkaloids. Vinca alkaloids are a subset drugs obtained from the vinca plant. They are naturally extracted from the pink vinca plant and they have been used to treat some diseases as diabetes, high blood pressure, and have been used as disinfectant. Vinca alkaloids are also important for being cancer resistance. There are 4 major vinca alkaloids in clinical use as vinblastine, veronil, vincristine, and vindycine. Vinca alkaloids are from most used class of cancer drug and will stay among the original and natural cancer therapy. The alkaloids that we use it as anti-cancer drug is vinblastine and vincristine. We started our work. We started our work in the lab with explant culture has been done on a mixed medium containing different concentration from growth regulators, where the different effects of growth regulators were studied when different parts of the plant have been cultured from leaves and stems. This is to induce callus formation and encourage growth. And the concentration that were prepared is benzyl adenine and naphthalinic acid and indole acetic acid. And the last one is 2.4-D. It was obtained the highest weight and the best variety from callus with using hormones, benzyl adenine and 2.4-D. After incubated it in the dark. And this was the first part of my research. And now I will be analyzing the alkaloid extracted from callus tissues using HPLC device to get the results. This is was last callus in formation. This is my reference. God willing, I will complete my master's degree on medicinal plant research. In the end, I thank all the attendants. Thank you for listening. This is my contact information. And thank you.
Thank you, Dr. Yusuf. I have a question, and my question would be, are you planning to analyze further these calluses that you produced in the dark, or what will you do next with your tissue?
Can you read this question?
Do you hear me? So, are you planning to do some analysis of callus tissue that you produced. What will you do with this callus later on?
Yes, my analysis I going to do it is using HPLC device for target vincristine and vinblastine alkaloids. Can you hear me?
Will you target them? Will you extract those alkaloids or what?
Yes, I will.
Are you planning to measure the concentration of it?
Yes, I plan to extract this alkaloids and know how this contains from it. Because I want to get a lot of this alkaloids from simple callus.
Okay, I think we will have to discuss it in chat perhaps or email, because I think you're losing the connections. I'm hearing you not that. I don't know for other listeners if they have any questions, comments. Can they hear it better? If no, then I will thank you for your presentation, a lot of colors, and at the beginning it was like a review paper on plant biotechnology, which was really useful, at least for me. So nice, and yeah, thank you very much.
You're welcome.
Thank you, Mr. Yusuf, for your nice presentation. So, and we just move on to the next presenter. Praise Ms. Carolina. Ms. Carolina done her bachelor degree in food science and technology and master's degree in food product technology. Her working experience is in an international food processing company, as well as a junior researcher and project engineer experience at the university. So she has done both industry and academic knowledge gained through the last 20 years provides a possibility to combine science and business in order to develop novel products enriched with bioactive compounds by valorization of byproducts from food industry to ensure a sustainable agri-food chain. Ms. Carolina, you're most welcome to begin your presentation now.
Hello, everyone. Thank you for introducing me. And today I will, can you see my screen?
Yes, we can see.
Can you hear me?
Yes, we can hear you and we can see your screen.
Okay, so hello everyone, and I am happy to participate in this conference, and I am glad to have an opportunity to present the results of my latest research on the topic application of ultrasound for cornstarch processing byproducts pretreatment and enzymatic hydrolysis intensification. Introduction, current trends in European Union agriculture are focused on the effective use of renewable sources into the development of novel biostimulant formulations. And one major group of plant biostimulants is protein hydrolysates, and they are defined as a mixture of peptides and amino acids. And a number of studies confirmed that biostimulants based on amino acids have a significant impact on plant growth, as they are very important for the metabolism of seeds. Also, they are used for the synthesis of seed storage proteins and, of course, as an energy source. Traditional options to produce this type of biostimulant is the use of biocatalysis to transform residual feedstocks into high-value target protein hydrolysates. For example, microbial proteases such as alkalis, flavozyme have been used to hydrolyze corn gluten meal. But the main disadvantages of traditional hydrolysis are long duration and a low degree of hydrolysis. And the majority of studies have been attributed to develop methods to improve the substrate conversion efficiency and reduce the time of hydrolysis. And ultrasound treatment is a novel technology which can be used to improve the extraction process of bioactive compounds, as well as assist the enzymatic hydrolysis. Can you see my slides good? Sorry.
Yes, we can see your slides.
Yes.
So this study was dedicated to investigate the effect of ultrasound pre-treatment at different frequencies of ultrasound on plant-based protein hydrolysis. As a feedstock proteins, corn wet milling byproduct known as corn steep liqueur was used, and 3 types of enzymatic hydrolysis have been carried out. So it is traditional enzymatic hydrolysis, low frequency ultrasound assisted enzymatic hydrolysis, and high frequency ultrasound assisted enzymatic hydrolysis. And the biostimulatory effect of obtained protein hydrolysates based on amino acids was tested on wheat seed germination characteristics. So ultrasound treatment has been carried out using 37 kilohertz and high intensity ultrasound and 850 kilohertz and low intensity ultrasound. And the efficiency of corn steep liqueur ultrasound treatment was characterized according to the soluble protein content determined by Bradford method. Then untreated corn steep liqueur and ultrasound treated corn steep liqueur has been enzymatically treated. And for the hydrolysis, concentrated liquid alkaline protease was selected, and the hydrolysis was carried out using different substrate concentrations, different enzyme concentrations, and different pH. In order to describe the efficiency of hydrolysis process, hydrolyzed protein concentrations was determined according to pH stat method, as well as kinetic parameters were calculated, and total free amino acid content was determined by ultrafast liquid chromatography. And finally, the biostimulatory effect of hydrolyzed corn steep liqueur was evaluating using wheat seeds. And after 3 days of the treatment, the germination percentage was calculated. And after 7 days of the treatment, stem and roof length, as well as stem and roof fresh and dry weight were evaluated in order to describe the germination study. And now I would like to present the results of my research. Here you can see changes in soluble protein content after ultrasound treatment. And the study showed that ultrasound power at low as well as at high frequency significantly affected the solubility of proteins in corn steep liqueur. And this study showed that by increasing the intensity of the ultrasound, the soluble protein content also increases. And the highest protein solubility can be achieved by using low frequency and highest intensity of ultrasound, and the same tendencies are noted by using high frequency ultrasound, as the highest solubility of constipated proteins can be achieved using also highest intensity, but the soluble protein content is slightly lower comparing to the low frequency ultrasound treatment. And at this stage of the experiment, low frequency, high intensity and high frequency, high intensity treatment were chosen for the further experiments. And now you can see the enzymatic hydrolysis of corn steep liqueur results with different substrate concentrations. And the results show that ultrasound pretreatment enhances the hydrolysis rate, especially at higher protein concentrations. The results also indicate that traditional hydrolyzed protein concentration can be remarkably improved in the beginning of the hydrolysis. It is after 20 minutes of the hydrolysis. And the highest protein concentrations were detected after 80 minutes of hydrolysis in the samples in traditional hydrolysis and ultrasound pretreated hydrolysis. But ultrasound increased the hydrolyzed protein content about 70% compared to traditional hydrolysis. In the perspective to determine the optimal conditions for enzymatic treatment, different enzyme concentrations were selected to test. And the results show that using traditional hydrolysis, the highest hydrolyzed protein concentration can be obtained after 80 minutes of hydrolysis using 5 grams per liter of enzyme. And high-frequency ultrasound pretreatment accelerated the hydrolysis, and the maximum hydrolyzed protein concentration was achieved after 60 minutes of hydrolysis using the same amount of enzyme, and low-frequency ultrasound pretreatment resulted in the highest hydrolyzed protein concentrations compared to all the samples using the lowest amount of enzyme. It is 2.1 gram per liter, and the shortest hydrolysis time, it is 60 minutes. And also the experiment has been carried out using different pH, this is 7 and 9. And as we can see from the figures, the tendencies are that with increasing pH, the hydrolyzed protein concentration increases in all the samples. And in all cases, the highest hydrolyzed protein concentration can be obtained at pH 9. And ultrasound treatment can increase the final HP concentration on average by 70.3%. Moreover, to confirm the ultrasound treatment effect on the hydrolysis, the kinetic parameters were calculated. And as you can see from the graphics, the initial hydrolysis rate increased on average by 36% with low frequency ultrasound pretreatment. And as well as Michael's Menten constant can confirm the ultrasonic treatment effect. This constant indicates the affinity of an enzyme for a given substrate. So the lower this value is, the higher the affinity of the enzyme for the substrate is. As you can see from the graphic, a major decrease of Michaelis-Menten constant on average of 33.5% was noted for the enzymatic hydrolysis with low frequency ultrasound pretreatment. So analysis of the application of alkaline enzymes into corn steep liqueur media confirmed that the concentration of enzyme preparation as well as the treatment time itself play a key role in the hydrolyzed protein content. And this study proved that ultrasound treatment using low frequency at the highest power could be an alternative approach for improving enzymatic hydrolysis process and increase corn starch byproduct protein solubility. At this stage of the experiment, the influence of ultrasound treatment at different frequencies on free amino acid content was compared. And the figure shows that positive changes in the total amount of free amino acid were obtained. This is corn steep liqueur untreated. This is treated only with enzymes. This is with pre-treated with low frequency ultrasound, and this is pre-treated with high frequency ultrasound. And the results show that application of ultrasound pretreatment for corn steep liqueur resulted in the higher total free amino acid yield, which also has a relationship with hydrolyzed protein content. And by using ultrasound pretreatment, on average, a 2.2 times higher free amino acid content can be achieved compared to traditional hydrolysis. At the final stage of the experiment, obtained protein hydrolysates were applied for wheat seeds coating to determine the germination study. Wheat seeds were coated with 10% of corn steep liqueur protein hydrolysate solution, and it was left for 30 minutes so the solution could soak the seeds. And the method was determined using roll method of the germination study, and the germination percentage was calculated after 3 days of germination. And as you can see from the results, the application of biostimulants increased the germination percentage on average by 9.4%. And besides, during the experiment, significant effects on wheat seed root and stem length were observed based on amino acids concentrations. And ultrasound treated wheat resulted in, on average, by 30% longer stems and longer roots. Also, treatment with biostimulants resulted in higher, for on average 24.2% higher biomass content of wheat stems and roots dry mass. So finally, I would like to summarize the experiment that corn steep liqueur is an inexpensive potential source of nitrogen to produce protein hydrolysates. Our study shows that corn steep liqueur sonication could be an effective way to accelerate the hydrolysis process and enhance protein solubility. When combining ultrasound treatment with hydrolysis, low frequency ultrasound pretreatment led to the highest hydrolyzed protein concentration using the lowest enzyme concentration and the shortest hydrolysis time, as well as low-frequency ultrasound sonication resulted in the greatest increase in total free amino acid content, and a significant increase of root and stem length, as well as root and stem biomass were measured when applying sonicated and enzymatically hydrolyzed products. And finally, ultrasound could be a sustainable way to include non-traditional methods to produce bioactive compounds in the agriculture industry. And thank you for your attention.
Thank you, Carolina. It was a great presentation. I personally liked it a lot, especially because it's on the ceilings, first of all, similar to our investigation. And yeah, I have some questions. First,
yes.
So you used these liquids as biostimulants, and you have seen that you got almost 25% higher mass of stem and root. Are you planning now further to analyze chemical profile of these wheat seedlings that you've seen? They have greater biomass. Probably they have changed their phytochemical content as well. You know, you gave them some proteins, some amino acids. They could definitely induce some changes in phytochemical content of your wheat seedlings. Are you planning to do some analysis later on?
We are more focusing on the technology of producing biostimulants, like to use ultrasound treatment. And now we are more planning to use the same material, the same protein hydrolysates for the foliar application, to check the foliar application to protect plants from abiotic stress. It was so the investigation of the phytochemical metabolites of wheat germs is not really my field. But if there would be an opportunity,
it's not in your interest.
No, it is, but we have no technique, I think, to do that for now. But if there would be an opportunity, I would like to. And also, I am searching for the, not searching, I'm interested in the new, not method, but the term, like biofortification. That you can improve the quality of crop by treatment, not only by genes, but also by treatment with some substances. So I am thinking about that.
Further analysis, because then that's a good thing to know, because then there is a possibility to collaborate with us, for example, because we are a laboratory for phytochemistry, and for us the analysis of phytochemical plants are something that we do on a daily basis. And obviously, I think I'm quite sure that your plants that you have seen now have higher biomass, stem and roots. They will probably, perhaps, I just presume, but we can test that. They will also have a higher concentration of certain bioactive components, you know, because you probably did not just improve their mass, probably you have improved their bioactive component content as well. And this is really interesting then later on for pharmacy or food industry. So if you are interested in some collaboration or some, if you can, for example, just lyophilize or dry your material, you can send it to us, and then we can then make the analysis of it.
Okay, thank you very much.
You know. We did some, just reach me per email. I will write my email here in the chat. So you can
Okay.
have it. And also I would just advise to do the statistics.
Yes, I am working on that.
It's always helpful. It's always
Yes.
good to have, but without statistics, you cannot publish it easily. So I think we could get us the results here. I'm quite sure about it. So yeah, I will put my email in chat, and yeah, you can reach me. Yeah.
Okay, thank you very much
So
for your
if,
questions
anyone
and
else
for your
has-
interest.
Yeah, you're welcome. Yeah. Does anybody else has a question for Carolina? If not, then we can go on further. Yeah, thank you, Carolina, once more.
Oh, it's so Krivana. Oh, thank you so much for the nice discussions as well. So, and we will be having a refreshment break now. Um, so we are ahead of 30 minutes. So we will be gathering again in 30 minutes. So we will have just a refreshment break or a health break for 30 minutes. Yeah, hello, Dr. Ivana, is it okay that we will gather in 30 minutes from now? Yeah, it's okay. Yeah, we will be having a refreshment break for 30 minutes. So get your refreshments and we'll be back in 30 minutes. Thank you. So welcome back everyone, and hope you had a very your refreshments and a health break. So and without any further ado, I will go for the next presenters. So Dr. Iqbal Hussain. Dr. Hussain has been working as tenure associate professor in the Department of Botany at Government College University, Faisalabad, Pakistan. Earlier to it, he had served this university as an assistant professor and assistant professor under HEC's interim placement of fresh PhD programs. Dr. Hussain, you're most welcome to begin your presentation now. Dr. Hussain, can you hear me? Yeah, hello. Dr. Hussain, can you unmute your microphone? We are unable to hear you.
Hello.
Yes, Dr. Hussain. Yeah, we can hear you now.
Okay. I will share the presentation.
Yes, you can do that now.
Okay. I have sent the presentation to Anthony, maybe to.
Okay, so just let me find that quickly so that we can share that.
Okay. Okay, I will share presentation. Okay. 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 You have clear? Hello?
Yeah, hello.
Now sharing?
So do you want us to share it or you're
Yes.
going to?
Yes. I have shared the slide.
Okay, you're going to share your slides, right?
Yes.
Okay, you can do that. Okay, no worries.
Okay, everybody has seen the slides?
So not yet, we cannot see, but it's still loading. Maybe it takes some time.
Okay. I have loaded.
So can you please double click on that? So maybe it is taking some more time to load.
Okay. Everybody has shown my slides?
So I think maybe Luis, my colleague,
Okay,
he started sharing the screen.
okay.
Is that right? Okay.
Okay, thanks so much.
Okay.
Lead-induced modulation in growth, chlorophyll pigment, nutrient uptake, antioxidant enzyme regulation, gene expression, and fruit quality into tomato cultivar. Next, please. Next, please. Next, please.
Yeah, hello.
Next, please.
Just a moment. So the slides were sharing, but it is not displaying on the screen. Just let me check with that.
Okay, I am sharing. Okay.
Yeah, okay.
Okay.
Yes, that's fine. Yeah.
Oh, that's fine. Okay. Lead-induced modulation in growth, chlorophyll pigment, nutrient uptake, antioxidant, enzyme regulation, gene expression, and fruit quality in 2 tomato cultivar. Heavy metal have become a severe heavy metal become a severe threat to ensure food security by affecting about 1/3 of the irrigated land on the earth. Heavy metal is among most drastic limiting abiotic factor to agriculture yield around the world. Stresses trigger a wide range of plant response in the term of cellular metabolism, alter gene expression, and changes in growth rate and crop yield. This picture indicates that maximum lead produced in Europe 28.7%, in USA 23.8%, and in Americas 7.7%, and Oceania 4.2%, in Eastern Bloc 18.2%, and in Japan 5%, and minimum lead produced in Africa 2.6%. World produce a lead about to 5.4 million tons. There are the different sources of the leads that increase the lead production in the soil and environment: urban soil waste, exhaust of automobiles, chimneys of factories, melting and smelting of ores, additives in pigments and gasolines, effluent from storage battery industries, fertilizer, pesticide, metal plating and finishing operator. All these sources that increase the lead production in the soil and environment. How metal affect the plant growth? When plant grow on the land, the plant absorb the lead from the soil and retains most of part of the lead in their roots. And they reduce the uptake of calcium and phosphorus and exert a adverse effect on the morphology and growth of the plant, and also reduce the photosynthetic capacity and inhibit the root and stem elongation and leaf expansion. Lead toxicity that disturbance in mitotic and abnormality, and also discharge our nuclei, and may cause the disorder in DNA synthesis and produce oxidative stress of chloroplast. Lead that interfere the enzyme which is necessary for the metabolism, that interrupt the growth parameter and photosynthesis alteration, transpiration rate changes, and reduce the germination, seed germination, and leaf chlorosis, and changes in the ultracellular structure. Tomato, Solanum lycopersicum is the botanical name. Tomato is economically important crop plant, can grow under all climatic region of the world. Tomatoes serve as a rich source of the mineral, vitamins, anthocyanin, lycopene, which have a capacity, which have a ability of anti-cancer and antioxidative properties. In Pakistan, tomato is a secondary primary vegetable crop. Nevertheless, ecological stresses factor that limit the tomato production. Metal, particularly lead, that harshly hinder the production of the potato. Hypothesis: lead stress might differently modulate the key physiological, chemical, and molecular process to affect the growth and fruit quality in the tomato. Objective to assess how different lead level stress affect the uptake, translocation of nutrient, deteriorate the fruit quality, and to find out the contribution of catalase gene in overall reduction of the lead induced oxidative stress, and subsequent subsequent role in altering the fruit nutritional value. Whereas the cultivar that shows greater catalyst gene expression and have a low hydrogen peroxide MDA content tolerated lead stress effectively. Plan of work, this study was conducted in the experimental research site in the Department of Botany, Government College University, Faisalabad during the winter season 2018, November 2018 to March 2019. Seedling of both up to 2 cultivars are selected on the basis of differential metal tolerance capacity. Nagina, that is tolerant, and Roma, that is sensitive, which are collected from the Research Institute Ayub Agriculture, Faisalabad. Plants were grown under light and dark period, 11 hour and 13 hour and 24 Celsius to 12 Celsius day and night temperature, average relative humidity 54%, but power value, photosynthetic active radiation value 687 to 948 macromole per meter square per second during the whole study. At the vegetative stage, the plant was subjected to different lead regimes, 160, 320, 640, and 1280 macromole were used for 3 weeks. For this purpose, a lead solution was prepared in a solution in Hogland solution and applied to the each parts that contain the plants in the increment of the 40, 80, 160, and 320 macromole each day until the desired level of the lead was achieved. And lead nitrate, which are used as a lead source. Experiment was laid out in completely randomized design with 3 replicates. The data obtained was subjected to two-way ANOVA using the COSTAT software. Different data were recorded, growth attributes and physiological parameters and biochemical parameter, catalyst gene expression with RT-qPCR analysis, estimation of nutrient uptake from the tomato plant, and fruit quality determination results. These bar graphs that indicate that when exposure of the tomato plant to the lead stress 160 to 1280 produce a considerable decline in the various morphological attributes, including the shoot, root, dry weight, and shoot length and leaf area. But response of the both cultivar were diverse regarding to shoot and root dry weight. Shoot dry weight was reduced in both tomato cultivar. Roma cultivar that shows more decline than Nagina under the different regimes over the control plant. Lead stress that decrease the root dry weight in both Nagina and Roma cultivar. But in Roma cultivar, this reduction is 50%, but in Nagina, this reduction is 39%. Lead stress inhibited the elongation of the shoot growth was more evident as compared to control plant, although Nagina shows less root length reduction than Roma. Lead stress that reduced the leaf area in both Nagina and Roma. But in Nagina, 52% were reduced, but in Roma, 54% were reduced over the control plant. Lead stress induced a visible decline in dry weight, shoot length, and leaf area in present study. This lead-induced decrease was more evident in Roma cultivar. Chlorophyll photosynthetic pigment, that is chlorophyll and carotenoid content, lead regimes differentially modulate the chlorophyll content and total carotenoid in both Nagina and Roma cultivar. Although two cultivars that shows the similar trend, but degree of the increase and decrease in the photosynthetic pigment are not the same. The change in the degree of decrease, the increase in the Roma was significantly higher than that in Roma, significantly higher in Roma than that in Nagina. Lead stress significantly reduced the chlorophyll A content, about 11 to 37% in Nagina and 5 to 41% in Roma. While lead regimes that reduce the chlorophyll B content about 9 to 37% in Nagina and 8 to 42% in Roma. In addition, total chlorophyll content about 10 to 37% were reduced in Nagina and 6 to 41% in Roma. However, the reduction in the chlorophyll content was higher in Roma than that in the Nagina. In contrast, the response of cultivars was similar regarding the total carotenoids. Between two cultivars, lesser content of total carotenoids were recorded in the leaf of Nagina under the lead stress. Biochemical attributes, oxidative stress, reactive oxygen species MDA or H2O2, ascorbic acid, and total soluble protein. Lead stress enhance the membrane disruption because of higher value of the MDA content, malone dialdehyde content. When tomato plants were exposed to the lead stress, lead regimes 160 to 1280 macron meter, the MDA content was increased in both Roma and Nagina cultivar, but these increases were maximally in Roma as compared to control. A significant increase in Nagina and Roma was also recorded under the different lead regimes 160 to 1280. A substantial decrease in ascorbic acid level was occurred in tomato plant when subjected to lead stress in growth media. In this case, toxic effect of the lead stress also triggered the significant decline in ascorbic acid content in Nagina and Roma, but this reduction was more apparent in Nagina. Total soluble protein content was significantly affected due to the lead regime stress. Lead stress markedly increased the total soluble protein in Nagina and Roma plants over the control. This enhancement was more in Nagina. Next, enzymatic, antioxidant, and catalyst gene expression. After lead treatment on tomato, the activity of antioxidant enzymes of both cultivars were first increased, and a catalase, peroxidase, ascorbate peroxidase were increased and then decreased. These changes were different between the cultivar. The plants of Nagina cultivar that shows increase in catalase activity, while Roma is superior in this contest during the various lead regimes. Lead stress markedly increased the APX content in Nagina, about 60 to 166%, and in Roma, this amount 45 to 135% over the control plant. In this study, we have recorded lead regimes cause a considerable declines in both Nagina and Roma cultivar. Maximum declines occur in in Roma as compared to Nagina. Compared with control, in the case of transcript level of catalyst gene expression, compared with the control after lead treatment, about 320 macromolar, the relative expression of catalyst gene was increased in Nagina and in Roma, but degree of change in Nagina is greater than Roma. Next, fruit quality of the tomato. Different fruit quality, moisture content, total fiber, fruit ash, fruit proteins. The lead stress affected the quality of tomato fruit. Lead regimes 160 to 1280 macromolar induce a steady decrease in moisture content in Nagina and in Roma. But ash content was increased more significantly. Ash content was increased more significantly in Nagina when subjected to the lead stress. A dose of, in the case of total fiber, a dose dependent lead toxicity that declined the total fiber in Roma and Nagina under differential lead regimes. In Nagina, 3% to 19% reduction, but in Roma, 0.5 to 5% reduction occurs. Lead stress markedly increase the total protein content in the fruit. In the fruit of Nagina and Roma. Nagina 7 to 35% increase occur, but in Roma 5 to 23% occur. In this context, higher fruit ash content, protein content were observed in Nagina, while in the Roma was inferior in this regard under lead stress. Glucose content and fructose content, fruit dry weight and fruit fresh weight. Glucose content was increased in Nagina 4 to 5%, but in Roma 9 to 14%. Under 320 and 1280 macromolar lead regimes. Fructose content was also affected, fructose content significantly affected due to the lead regimes. Higher concentration of fructose content, about 14%, was recorded in Roma plant at 1280 macromolar. In this context, higher fructose and glucose content were observed in Nagina, while in the case of Roma, these characteristics, these fructose and glucose contents was inferior in this regard under the lead stress. Fruit dry weight, lead reduced the fresh and dry biomass of tomato fruit. Nagina cultivar shows maximum reduction in fruit fresh and dry weight than Roma under different lead regimes. Next, mineral content in the tomato plants, we say it nutrient uptakes, sodium content and potassium content and fruit calcium content. The response of both cultivar. In the case of mineral contents, was inconsistent regarding the sodium content. Lead stress subsequently increased the sodium content in the fruit of Nagina, about 3 to 7% at 160 to 1280 macromolar, respectively. This increase was recorded 6% in Roma at 320 macromole. In contrast, the lead toxicity increased the potassium content significantly in the Roma, about 7 to 20%, and then that Nagina 1 to 15% under higher lead level, about under higher lead level such as 640 to 1280 macromolar. In the case of calcium content in the fruits, calcium contents were reduced in response to lead toxicity. This reduction was being more maximum in Roma, about 13 to 26% at 160 to 1280 macromolar of the lead stress. Lead uptake and accumulation in the root and shoot, leaves, and fruit of the tomato. After application of the lead treatment, 160 to 1280 macromolar, compared with the control, accumulation of lead content take place in the root, shoot, leaf, and fruit were more significant in tomato plants under the lead stress condition. Lead content were increased more in the root, then shoot, and then leaf, and of the both Nagina and Roma cultivars under the different lead regimes. After lead treatment, lead accumulation may also occur in the fruit of both Roma and Nagina cultivars. Results indicate that the significant amount of lead accumulation in the root compared with its concentration in the shoot, leaves, only small amount of the lead that reaches to the fruit. Conclusion, the result indicated a significant reduction in growth attributes, photosynthetic pigment, yield attributes, calcium ions, uptake in tomato cultivars under lead stress, although damaging effect was more evident in sensitive cultivars. Moreover, the lead stress enhanced the oxidative damage in the term of higher MDA H2O2 content in tomato plants. However, the lead stress decreased the fruit quality, whereas the tolerant tomato cultivar show upregulation of catalase gene expression and had less MDA and H2O2 content, that is ROS content, reactive oxygen species content, thus show higher lead stress tolerance than sensitive one. Further, the tolerant cultivars show better antioxidant system, lesser degradation of the chlorophyll, and better growth under lead stress. This is the reference. Thanks so much for attention, please. Thanks so much for attention, please.
Thank you, Professor Iqbal. You know what makes me happy about your results? Now, at one of the last slides, you have shown the concentration of lead in root, leaves, fruits, and then I saw, okay, in root, really high concentration, like 200 micromole per gram of dry weight, and in fruit, it gets just like 4 micromole of per gram of dry weight. That makes me happy. At least it's not transported that upwards to the fruits. And I actually got an idea. It would be nice to, I mean, we cannot clean the soil from the weed, but perhaps we could just stop the transport from the root upwards. So at least we will be able to use fruits without weed. That's just a funny idea. Does anybody has a question for Dr. Iqbal? Then thank you once more, and we can proceed further.
Yes, thank you, Ivana, and thank you, Dr. Iqbal Hussain, for your nice presentation. So, and we'll move forward for the next presenter, Professor B.N. Hazarika. Professor B.N. Hazarika is presently working as a dean at the College of Horticulture and Forestry, Fasigat, Arunachal Pradesh. Professor Hazarika guides a number of PG and PhD students and handles several externally funded research projects and organized 90 trainings. He has published 80 research papers, published 20 books, 25 conference papers, and book chapter, learned practical manual, 25 patents, edited 13 seminar, and 245 popular articles. He contributed significantly in collection, morphological, and molecular characterization of diverse genotype of various fruit crops, standardized good agricultural practices for some major fruit crops, introduced new fruit crops in the region. Professor Hazarika, you're most welcome to begin your presentation now. Thank you so much.
Thank you, sir. Thank you. I am now going to share my slide.
Yes, you can share your slides.
Is it visible, sir?
Yes, your slides are visible. Please go ahead.
Okay, thank you very much. First of all, I thank the organizer for giving me this opportunity to be with you in this very nice program. So I am coming with one of the very important topic of this presentation, that is acclimatization of in vitro cultured plants for in vivo conditions. So dear expected viewers, I'm going to discuss this topic on the basis of my whatever the works that is being done on this aspect. And already it is being published in the very referred journals. So for example, this is published in the Current Science about the acclimatization of the tissue culture plants. And this is also published in the Scientific Horticulture about the morphophysiological disorders of the in vitro cultured plants and how this methodology of hardening is taking place in case of in vitro cultured plants. So this has already been published in the Scientific Horticulture. And the similar works on effective acclimatization of the in vitro cultured plants, the methods, physiology, and genetics are also published in the Global Science book from UK. So with this, I'm now going to present my presentation. As you know, the in vitro propagation is nothing but your, there are various kind of propagations are there. For example, micropropagation, a meristem culture, somatic embryogenesis, soma-clonal variation, embryo culture, in vitro selection, enter culture, and photoplast culture. These are the various types of in vitro propagation, as you know. So, as you know, the micropropagation, as you is done for the rapid propagation of the superior plants while maintaining the genetic makeup. And meristem culture is also taking place for elimination of the disease-free planting material. And somatic embryogenesis is done for rapidly increasing the desirable plants while maintaining the genotype of the original plants. Similarly, the soma-clonal variation is also used to induce the desirable heritage changes in the regenerated plants. and embryo culture is done to rescue the embryos during the attempt in the case of hybridization. And interculture is also done to produce the homogeneous and pure breeding lines of the plants and hybrid production and genetic studies. And protoplast culture is generally done for incorporating the potentially useful genes. So with this, I'm now going to the need of the acclimatization. Why the in vitro propagated plants need acclimatization or acclimation and before the transferring of the tissue cultured plants or in vitro cultured plants to the in vivo condition, that is the outside the lab. And actually what happened when the aseptic culture of the plants are grown in the in vitro method, that leads to the development of the culture induced phenotypes. So what is culture induced phenotype? This phenotype is basically, this is a very delicate plant and susceptible to the transplant shock because these plants were grown in the low level of light, aseptic condition and medium containing sugar and high level of humidity. So these are the factors for which contributed to development of the plants which are called culture-induced plants phenotype, and that those plants are not getting hardened unless these plants are getting hardened, they are not possible to make it success in the outside conditions. So with this objective, there is always the need of the acclimatization or hardening of the in vitro culture plants. So in general, what happened in vitro regeneration, actually what happened, these are the normal procedures in case of micropropagated plants or in vitro culture plants. For example, when we are taking the explains, this is nothing but your undifferentiated callus and axillary bud proliferation and organogenetic callus, and from there multiple shoot is being initiated, and then elongation and rooting, and finally the acclimatization takes place. And these are the normal protocol for the tissue culture plants. So for this, when we are going for these tissue culture plants or in vitro grown plants, the explains may be of various types, depending on the types of the plants, and the physical environment, that is culture condition, is medium pH and temperature and light is very important. And there are various type of media formulation, for example, MS media, B5 media, and other media combined with the plant growth regulation, regulators, and various sucrose concentration and gelling agent like agars. So as you know, the organogenesis is, as you know, the plants are starting the formation of the callus. And if we are going for the multiplication of the callus, definitely it will go for proliferation. So similar kind of works are being done in citrus crops as well as in some carnation plants. We have done it. And similarly, some of the crops, that is bale, jelly marmelos, you see how the morphogenesis takes place in case of bale. So these are some of the works in some other medicinal plants. So during the process of rooting, the microshoots, when the rooting hormone is added to the media, then rooting takes place, and that is why it is being done. And in case of efficient method of acclimatization of the micropropagated plant in case of citrus crops has already been done in our earlier works, and in case of citrus it is being published. So in case of culture in this phenotype, as I have told earlier also, the aseptic culture of the plants by in vitro method, they lead to the development of the culture-induced phenotype, which is because of the low level of light, aseptic condition, and medium containing sugar and high level of humidity. And because of these favorable factors, make the tissue culture plants or in vitro grown plants are susceptible to the transplant shock. And when we are getting it transferred to the greenhouse condition, they are subjected to the transplant shock, and loss of plants takes place, survivability is less. This has already been published in Indian Journal of Horticulture. So the factors, in case of some of the plants, another this bottleneck is that your hyperhardicity. So what is hyperhydricity? That hyperhydricity is a morphological and physiological disorder of the plants vegetatively propagated in vitro. This is a very classical disorder, or you can say in physiological disorder of the in vitro grown plants. And plants, what happen? They become the glassy appearance, and their stems and leaves are often thick, rigid, and easily and decrease protein with the chlorophyll content. And this is the case in case of carnation that we have done and published in this some of the journals like Indian Journal of Horticulture as well as Acta Horticulture. It has been published. So you see, in case of normal plants and these proliferated plants, these are some of the under the hyperhydrated condition. It is in case of carnation plants, these are the normal plants, and these are the hyperhydrated plants under tissue culture vessel. So the anatomy also somewhat making difference, and we have studied in case of citrus plants through the scanning electron microscope, which has been already published in the Indian Journal of Horticulture. What happened in case of anatomy, the large vacuolated mesophyll cells, they are there in case of in vitro culture plants, and hypertrophy of these cells and the large intercellular spaces, a lack of epicuticular wax, and chloroplast presented abundant plastic globuli, and guard cell were different in their morphology, and also high level of the potash. So these are some of the characteristic features of the anatomy in case of in vitro culture plant, because of which the in vitro culture plants need acclimatization. And you can see the scanning electron microscopy photograph of the citrus plants, which has been also published in the Indian Journal of Horticulture during 2002. And here we can see the epicuticular wax is one of the important criteria for reducing the transplant stock in case of in vitro culture plants. And it is found that the measurement of the amount of the epicuticular wax on leaves of the culture plants revealed that a lack of the crystalline structure was correlated with the significantly less epicuticular wax compared to the greenhouse grown plants. So this is the scanning electron photograph. It is seen here that amount of the epicuticular wax produced in the in vitro culture plants are characteristically lack of the crystalline structure, and also less as compared to the greenhouse grown plants. So this is, you can see another criteria, that is your stomata become open. And here in case of normal cases, it is closed. And it is also being published in case of Indian Journal of Agricultural Research. It is because of the peclobutazole. And another some of the factor is your sucrose. As you know, sucrose is used for giving the energy to these nutrients to these in vitro culture plants. And what happened, the excess, the dose of the sugar is also influencing. And in case of some of the tissue culture plants, excess moisture in the culture vessel also taking place, and sucrose induced biochemical changes in the in vitro microshoots of the citrus plants also studied and published in this. And another is your influence of the agar. And you see, increasing the concentration of the agar in the medium also has a direct effect on the hardiness of the plants later produced. So another is these symbiotic organisms. In early inoculation with an appropriate symbiotic organism promises to improve the plant survival and performance. This is very interesting study. And another study we have done it your peclobutazole. As you know, peclobutazole, what happened if we are using in the growth medium, preconditioning microshoots with peclobutazole influence the higher ex vitro survival by intensifying the internode length and thickening roots and reducing leaf dehydration by regulating the stomatal function and increasing the epicuticular wax per unit area of leaf of the in vitro culture plants. This is very interesting one. because this paclobutazole is a growth retard, and when we are using it in case of growth medium, it is having this beneficial effect of like this, and also mostly the epicuticular wax play an important role in reducing the transplant shock by reducing the transpirational loss. So this is how the inclusion of the paclobutazole in the growth medium produce stomata with minimum apparatus, unlike the normal stomata due to the general reduction in the cell expansion that is caused. This is also published in the Folia Horticulture, published from Poland. So this is how we have done this work on this micropropagated plants and their hardening. This is the paclobutazole. Actually, paclobutazole, what paclobutazole does is, paclobutazole inhibits the curine oxidase and thus blocks the oxidative reactions from end-curine to end-curinitic acid in the pathway leading to the zebarlic acid. In short, paclobutazole is inhibiting the zebarlic acid production, and thereby the plant's growth is being restricted. And also it is published in the Indian Journal of Agriculture Research during 2002. And another study, that is your P-conditioning with the sucrose, you see the larger persistent leaf packed with greater amount of the storage compound would contribute more after transplantation. And increasing the concentration of the sugar in the medium maximize the nutrient function of the persistent leaf. That means the persistent leaves play an important role in the survivability of the in vitro cultured plants, and this can be regulated by using the sucrose. And this has already been done in published in the Acta Botanica Croatica, and which is published from Croatia. And this is another paper that is, you can say, another method, another strategy by reducing the humidity. With the reduced humidity, the plants developed under lower humidity will have fewer transpiration and translocation problem ex vitro, and persistently that looks like a normal. So this is how it is done by putting some silica gel in the culture vessel, and it will reduce the humidity, and reduction in the humidity in the culture vessel has this beneficial effect, like your persistent leaves will be looking more normal than this, the in vitro culture plants. And this has also been published in the Agrotropica, published from Brazil. So another very interesting is your photosynthetic efficiency. So in vitro culture plants are either poor in chlorophyll content or the enzymes responsible for photosynthesis. That is very interesting. And ribulose biophosphate carboxylase are also inactive or absent in case of in vitro culture plants. And this aspect of the photosynthetic system of cauliflower meristem cultures in vitro as compared to seedlings and plants are established in the soil are shown here. Another this strategy is your control of the carbon dioxide concentration in the media, in the cultural chamber. The control of the carbon dioxide concentration achieved in acrylic boxes and having an air tight lid on the top, and this is how increasing the carbon dioxide concentration also helping in hardening. Another very interesting is your, in case of citrus microshoots, we have done it, that is direct rooting and acclimatization of the citrus microshoots in carriers, different type of carriers. And here in this picture, it is the carrier is soil right. Here you can see it is the cutting of the microshoots are directly placed in case of carrier that is soilite with combination with the different type of medias. And what happened, they will give the root, and it will bypasses some of the steps of the rooting in the tissue culture vessel. This is what we have done, the direct rooting and acclimatization using 2 centimeter long microshoots of Azalia marmelos after treating them with IBA at 10 ppm for 2 minutes. And simultaneously, the ex vitro rooting and acclimatization was also achieved with using different carriers. So among the different carriers, soil right layered with the farmyard manure showed better rooting, that is 80 to 91%. and high ex vitro survival, that is 90 to 97%, which is why so important in the over the conventional routing in agar-based medium, and this has already been published in the in other journals. So this is also very interesting. And another aspect of the hardening is your photoautotropic micropropagation of the important horticulture and ornamental plants. And this is already been discussed, the improved gas exchange in culture vessel, that is enhancement of the carbon dioxide or reduction in oxygen, also increasing by increasing light and penetration of the container content and decrease the relative humidity in the vessel. These are some of the techniques used for the different type of the plants. So this is how these strategies are being discussed. And finally, I can come to the conclusion that the ultimate success of the in vitro propagation on commercial scale depend on the ability to transfer the plants out of the culture on a large scale at a low cost and with high survival rate. In vitro culture plants are generally susceptible to the transplant shock, leading to the high mortality during the final stage of the micropropagation. That is very upset matter. Understanding these abnormalities is a prerequisite to develop the efficient transplantation protocol. So this is my conclusion of my paper. And thank you for patience hearing. Thank you very much. And thank you once again for giving me the opportunity. Thank you very much to organizers.
Thank you for the nice presentation. Does anybody has any questions, comments? If no, then thank you once more, Professor, and we will go on further with the next presentation.
Thank you, Ivana ma'am. So we'll go for the next presentation, and who is Ms. Shweta Gupta. Ms. Shweta was an inspired scholar during her PhD in microbiology, which she completed from Dr. Ashwant Singh Parmar, University of Horticulture and Forestry from Solan, India. She has shown 2.5 years of working experience with plant growth promoting bacteria. Until now, she has published 12 research papers, 4 research articles, and 3 review articles. She has participated in international conferences and 3 international conferences, and she is an ICAR, SRF, and NAT qualified and have been awarded with Best Poster Presentation Award in national and international conferences. And currently, she is working as subject matter expert in biology with TREG Incorporation and American Education Technology Company. Ms. Shweta Gupta, you're most welcome to begin your presentation now. Thank you so much.
Sir, am I audible?
Yes, we can hear you.
Am I visible too?
Unfortunately, no, we cannot see you. We can see a blank image there.
Yes.
Yes.
Now I think I'm visible.
Yes, all right. Thank you. Yeah.
Let me share my screen first. Is it visible?
Yes. Yes, now it is okay. Perfect.
Okay, good afternoon to all. Myself, Shweta Gupta. First of all, I would like to thank to give me an opportunity to represent my work in this conference. And the topic of my presentation is plant growth promoting rhizobacteria and chemical fertilizers, their impact on soil health and productivity of capsicum. As we all know that environmental degradation has affected the human and animal health, therefore there is a need for increased productivity of the crop. that with the sources which are eco-friendly, non-pollutant, and cost-effective. So PGPR is among those technologies. PGPR, plant growth promoting rhizobacteria, that not only improves the soil physical, chemical, and biological properties, but also enhances the yield and are known to supplement the chemical fertilizers and pesticides. So I carry out the research where I isolated, screened, and molecularly characterized indigenous plant growth promoting rhizobacteria from the capsicum, rhizosphere, and root plants and studied their effect as biofertilizers, biostimulants, and biocontrol agents so that I can have the one inoculum for a specific crop from different locations to be effective in different locations. Sample collection was done from the different sites of agroclimatic zones 1, 2, and 3 of Himachal Pradesh, India. Isolation and enumeration of microbial population was carried out using the serial dilution plate technique, where the isolated microbes, they were further screened for phosphate solubilization and nitrogen fixation using the replica plate technique. These are the results. Among all rhizospheric bacterial isolation, maximum rhizospheric count was reported from agroclimatic zone of 2 of Himachal Pradesh. And for endophytic, the maximum endophytic count is reported from agroclimatic zone 1 of Himachal Pradesh. After isolating morphological different isolates, they were screened for various plant growth promoting activities. And among all 157 isolates, I had in hand 34 isolates with plant phosphate solubilization, nitrogen fixation, and pseudophore production efficiency. These 15 isolates, they were then tested for various plant growth promoting traits like phosphate solubilization on PVK medium, pseudophore production on CAS medium, and HCN production by the PGPR isolates. ACC deaminase by ACC deaminase production, nitrogen fixation, and IA production, they were also screened for the same ammonia production. After screening for plant growth promoting isolates, these are the results. The different bacterial isolates, they were screened, and all the bacterial isolates, they were positive for one or more PGP traits. After that, these PGPR isolates, they were further screened for biocontrol efficacy against the various pathogens of the capsicum, like Fusarium, Rhizoctonia, Pythium, Ralstonia, Phytophthora, and Clatotrichum. And all the isolates, they were having antagonistic activity against one or more fungus. However, none of the isolate was found to have effective biocontrol efficacy against all the tested isolates. Then after that, we screened the isolates for lactic enzyme production. And similarly, they were having some 2 or more of the lactic enzyme production activities. After screening, morphological and physiological characterization of the isolates was carried out, and they varied in form, elevation, and Graham's reaction. None of the isolate was able to grow at 45 degrees centigrade and pH 9 and salinity of 10. However, they varied with biochemical responses. After their molecular and PGPR characterization and selection, we carried out the field experiment where we selected 6 best PGPR isolates for seedling studies. And then among 6, 4 were selected with different chemical fertilizer doses for net house studies. And from net house studies, we selected the 2 isolates with different chemical doses for field studies for 2 years. These are the results of my seedling stages, where among all the tested isolates, isolate MAT8, KAN11, JHA6, and ROH40. They excelled over other tried isolates and they were further selected. This is the net house studies where it can be clearly seen that treatment 8, where we uses 80% recommended doses of NP fertilizers along with the isolate ROH14, it excelled almost in all the studied parameters and was statistically at par with treatment 7 and treatment 1. Similarly, in cases of root biomass and fruit length and fruit weight, this treatment 7 excelled was statistically at par with treatment 8 and treatment 1, that is control 100% recommended doses of chemical fertilizers. Similar results were reported when we studied the plant and nitrogen, phosphorus, and potassium content and their uptake. Treatment 7 and treatment 8, they were excelled and was found to be at par with the control. However, non-significant effect was reported on pH, EC, and OC of the soil. And when talking about soil, available nitrogen, phosphorus was also enhanced by the inoculation of the PGPR with reduced chemical doses, that is treatment 7 and treatment 8, as compared to the treatment 1, that is control 100% recommended doses of chemical fertilizers. So here we get an idea that if can be tried on the field, let us see whether the chemical doses can be reduced efficiently or not. These are the biological parameters under the NatHouse studies, and similarly, they were also enhanced as compared to the control with the 80% and PGPR inoculated. And it was quite interesting to report that, and quite obvious also, that when the PGPR, they are isolated in the soil as compared to their only chemical fertilizers like treatment 1 and compared with these chemical fertilizers of treatment 6, 7, 8, they had reported enhanced soil biological properties. Now, field studies were carried out with reduced doses of chemical fertilizers by 20 and 40%, and 2 isolates they were selected, that were GHA6 and ROH14, applied in single or in consortium. And it was amazing to record that treatment 4, that received the 80% of the recommended doses of NP fertilizers along with the consortium of the bacterial tried isolates, it reported the maximum studied plant parameters and was statistically at par with the treatments receiving the isolates singly at 80% doses and 100% recommended doses of chemical fertilizers. Similarly, results were reported for soil nitrogen and phosphorus content and soil biological parameters like microbial population and microbial biomass. Soil enzyme activities, they were also recorded, and similarly the treatment 4, that was statistically at par with treatment 2 and treatment 3, that received the bacterial isolates with 80% of the recommended doses of chemical fertilizers. Now, after studying the biofertilizer efficacy of the PGPR isolates, they were further screened by our team for the efficacy under the water stress conditions. These are the NatHouse studies, the general view, and water field capacity at 100%, 80%, and 60% and 40% was tested with the 2 bacterial isolates. And among the different isolates, 80% of field capacity and isolate are best isolated.
So I would like to request the participants that maybe Ms. Shweta Gupta, I think she had a network problem. So kindly wait for 1 to 2 minutes so that I think she may be rejoined us again. Dr. Ivana, I think Ms. Shweta Gupta had facing her network issues. So shall we go for the next presenter? So in the meanwhile, if she connects, we will proceed her in the next presentation, okay? Yeah, okay. So, oh yeah, she entered the room, okay? We will allow her to drop her presentation. Dr. Shweta Gupta, I think you have joined us. Can you please unmute your microphone? So your microphone was muted. Can you please unmute that? Yeah, okay.
Can you hear me?
Yes, we can hear you.
Sorry for there was some internet problem.
Yes, no worries, because it happens always because we're in the digital world and this happens sometimes. Okay, no worries. You can proceed with the presentation now.
Now is my slide visible to all?
Yes, it is visible. You can proceed with that.
I have to start from beginning again.
No. You can start.
I lost the connection. I was just speaking.
You can resume from the last one, okay?
Okay. So I was at my field, net house studies. I was telling you that treatment 7 and 8, that is 80% of the recommended doses of NP fertilizers, they reported significantly higher microbial biomass and population as compared to the treatments which did not receive any of the bacterial fertilizers. Now the field studies were carried out for 2 years, where we compared 80 and 60% recommended doses of NP fertilizers with the two best isolates, GHA6, ROH14, and along with the consortium. And it was quite interesting to record that the treatment 4 that receives 80% of the NP fertilizers along with consortium of the two selected isolates, it reported significantly higher plant growth and yield parameters and was statistically similar as compared to the bacterial isolates when applied singly as in treatment 2 and 3 with 80% recommended doses of NP fertilizers and treatment 1 that is 100% recommended doses of chemical fertilizers. Similar were results for available NNP content of the soil and the bacterial population and microbiomass of the soil. Now, effect on the soil enzymes was also studied, and it was interesting to know that the treatments receiving the PGPR isolates, they reported higher enzyme activity as compared to their respective control. That is, treatment receiving the bacterial isolates, like 80% of the PGPR isolates, as compared to the control, they reported higher soil enzyme activities, which is responsible for the higher productivity of the soil. Now, after testing for the biofertilizer efficacy of the tested isolates, we studied whether these isolates, they are effective under water stress conditions also. whether they can be effective as biostimulants also. So they were tested at 180, 60, and 40% of the field capacity. And it was recorded that 80% of the field capacity having ROH14 isolate reported maximum plant parameters, which was statistically at par with the treatment tree receiving 80% of field capacity with GHA6 isolate and 100% of the field capacity, thereby indicating their drought efficacy. And it is helping the plant to sustain the drought also, besides their biocontrol efficacy. Similarly, the results were for NPK content and their uptake. Drought efficiency is mainly attributed to the antioxidant enzyme activities. So they were screened for superoxide dismutase, peroxidase, and catalase activity. Treatments receiving the bacterial isolates, they reported higher of the antioxidant enzymes activity and therefore are responsible for higher plant growth or sustaining the plant growth under the drought conditions also. Now, after studying the biostimulant effect and biofertilizer effect, the best isolates were also screened for the biocontrol effect of the capsicum. But among all the pathogens, I studied Ralstonia solanarisum because there I got the success in infecting the plant with the pathogen. And it was recorded that RHO14 isolate, it reported maximum of the plant parameters, and ROH6 isolates when the plant has been infested with the pathogen Ralstonia solanarisum, and therefore the maximum biocontrol efficacy was reported for ROH6 isolate followed by ROH14 isolate. So I in hand have 1, 2, 3 isolates that have biocontrol, biofertilizer, biostimulant effects, and the studies are further going on for their multi-location trials. They were molecularly identified using the 16S ribosomal RNA sequencing technique, and the sequences have been submitted in NCBI, and they are identified as Pseudomonas aeruginosa, GHA6 isolate, ROH14 isolate, Bacillus amyloliquefaciens. These two were biofertilizers and biostimulants, and ROH6, which also have best biocontrol activity as Providentia species. I would like to conclude that the conjoint application of PGPR isolates at 80% doses of NP fertilizers has good aspects to be used as biofertilizer, as are known in partial replacement of NP fertilizers by 20%. Biocontrol, as reported, the biocontrol efficacy against Ralstonia bacillus and biostimulant, as reported, induced tolerance in the plants, and therefore not only enhance the growth and yield, but also helps to sustain the soil health in a longer term. These are my publications which I have in hand from this my research work, and I have also been awarded the 2 awards in national and international conferences. And thank you for listening, and also sorry that I lost some connection in between, and thanks again for the forum for giving me the platform to share my research work. Thank you. Hello? Am I audible?
Yes, hello.
Hello?
Yeah, hello, yeah. Thank you very much, Dr. Shweta Gupta, for your nice presentation. Thank you very much.
Thank you.
So, any questions for Dr. Gupta? So if there are no questions, so we'll move on to the next presentation. I would like to thank you once again, Dr. Shweta Gupta, for your nice presentation.
Thank you, sir.
You're welcome. Thank you. Okay, let's move on to the next presentation. Here from India. Dr. Narendra Kumar. Dr. Narendra Kumar and his PhD in 2002 from Gorakhpur University. Since then, he's doing research and teaching UG and PG students. And presently, he's working as an associate professor at Avanti Institute of Biotechnology, which is in Haryana, India. And he has attended and presented a large number of papers in several national and international conferences. Dr. Narendra Kumar, you're most welcome to begin your presentation now.
Thank you. Okay, thanks to organizing secretary for giving me time to present my work. Myself, Dr. Narendra from Amity University, Haryana. Whether I am audible?
Yes, you're audible. Yes, you're audible, and I would like to request the remaining participants to mute their microphones because that's really distracting the speaker to present his presentation. So, remaining others, I would like to kindly request all of this to mute your microphones, please.
Okay. The topic of my presentation is efficacy of clove oil, a plant product in enhancing shelf life of food seeds of pigeon pea. Loss of food commodity due to pest infestation is a major reason of food crisis. In spite of improvements in hygiene, it has been well estimated that almost 30% people in industrialized countries suffer from foodborne diseases each year. In 2000, 2 million people died worldwide. So new methods are needed for reducing foodborne pathogens, possibly in combination with existing methods. This has been observed. Industrialized societies appear to move towards green consumption, desiring fewer synthetic food additives and products without harming the environment. The versatile composition of essential oils, broad spectrum antimicrobial potential, and low toxicity enhance their widespread use in capable of pathogens and type of processed food. Prolong the storage life of foods by preventing rancidity and oxidation of lipids. The pigeon pea, also known as pigeon pea, red gram, tur or gungo peas, is a perennial legume from the family Fabaceae. Since its domestication in the Indian subcontinent at least 3,500 years ago, its seeds have become common food in Asia, Africa, and many parts of the world. It is consumed on a large scale in South Asia and is a major source of protein for the population of Indian subcontinent. It is the primary accompaniment to rice or roti, flatbread, and has the and breadth of India. World production of pigeon pea is estimated at 4.49 million tons. About 63% of this production comes from India. The total number of hectares grown in pigeon pea is estimated at 5.4 million. India accounts for 72% of area grown to pigeon pea, or this is around 3.9 million hectares. This contains 23% carbohydrate, 44% protein, and 2% fat. Pigeon pea is typically stored for 6 to 8 months after harvest, but more than 90% of farmers don't take any precautions to protect it. Its proper storage enables farmers to earn high margins. Most farmers sell their pigeon pea seeds in village, local, and urban markets. Approximately 20 to 30% of stored pigeon pea food seeds become deteriorated by fungi and insects. The non-scientific storage of pigeon pea in rural areas leads to heavy deterioration of pigeon pea seeds by fungi and insects. But detailed studies is still lacking. Their management is done through use of chemical pesticides such as phosphine and methyl bromide. Insect resistance to phosphine is a very global issue now, and controlled failures reported in field situation in countries. Methyl bromide, broad spectrum fumigants, declared as a ozone repleting substance and is being phased out completely. In view of the problems with current fumigants, there is a global interest in alternative strategies, development of chemical substitutes, exploitation of controlled atmosphere, and integration of physical methods. So among botanical extracts, botanical management is also an alternative. Among botanical extracts used as a pesticides, essential oils are promising alternatives to chemical pesticides. So the interest has been shown in plant products, that is essential oils, for fumigant action, since it is believed that natural compounds from plant sources may have the advantage over conventional fumigants in terms of low mammalian toxicity, rapid degradation, and local availability. The use of volatile antifungal substances for the control of diseases caused during storage has been recognized to have better future over other fungicides. Therefore, the present study was undertaken to search a renewable, biodegradable, non-phytotoxic, and harmless botanical pesticide for the management of deterioration of peas and pea. So that's therefore mycological analysis and samples of peas and pea were collected from grocery stores at Gurgaon City market around 25 places in India. They were examined for associated macroflora. Techniques adopted were agar plate method, that is musket, and blotter method. Total number of fungi here investigated was 16. During the blotter method of investigation, this was 14. Agar plate 10. Most dominant fungi were Aspergillus flavus and Niger. Only one species, that means Clasobricus chinensis, was found to be present. आगे क्यों नहीं जा रहा? ये रुक गया, लाइट चली. Okay, this is the fungus. This is the Niger, Aspergillus niger. Its dominant showing, its cordylia showing how this. So, only one insect beetle was there. That means pulse beetle. That is Clasobricus chinensis. The attack is during July to August in the store. The adult feed on the peas and pea by making a small hole. Infested stored peas and pea seed can be recognized by the white X on the seed surface and the round exit holes with the flap of seed coat. The adult stage is described as being brown in color with black and gray patches over the body. So the deterioration caused by dominant isolated fungi, Aspergillus flavus, Niger, Ochraceus anterius, and pulse beetle Clasobricus chinensis, with respect to weight loss and seed germination was evaluated. Aspergillus flavus and Niger species played a very important role in seed weight loss and seed germination. The protein content in Aspergillus flavus inoculated seed was 24%. In Niger, this was 25%. Clasobricus chinensis 26%, while control set without fungus showed 44% protein content. The carbohydrate content in Aspergillus inoculated was 10%, Niger 13%, Clasobricus chinensis 14%, while control set showed 23%. On account of wide occurrence and their pathogenicity, these were selected as test organisms. There was significant decrease in weight in treated sets of flavus, Niger, and insect. So these species also reduce seed germination also. So that's why 100 plants were screened through Clevenger apparatus. Their oils were extracted and they were evaluated against dominant fungi, Aspergillus flavus and Niger, and insect Clasobricus chinensis. The technique adopted was inverted petri plate technique. So that's why out of these 100 plants, only one plant was found to be highly active, that is clove. The botanical it is Syzygium aromaticum oil. Its MIC was found to be 300 ppm. Oil showed fungistatic action at 300 ppm. This was fungicidal at 400 ppm. And during a spectrum study, it inhibited 10 fungi at 400 ppm and 16 fungi separately at 600 ppm. So clove essential oil showed 100% instant repellent activity against Clasobricus chinensis at 0.0 ml dose. So this is the clove from where the oil was extracted. So the physical factors on activity oil were studied. Temperature treatment up to 100 degrees centigrade given. No adverse effect on the toxicity of the oil. Autoclaving was again having no effect. Storage period up to 120 days given no effect. Increased inocula was having no effect. 5 fungal disc of 5 mm and even 1 fungal disc of 11 mm. So physical chemical properties of clove oil were studied. These are the parameters having a specific gravity 0.9559, having a specific rotation plus 10, reparative index 1.450, acid value 3.5, ester number 150. Phenolic content was found to be nil. It was completely miscible with petroleum ether and acetone, and 90% ethanol ratio 1:1 ratio, but this oil was insoluble in water. GCMS analysis revealed presence of 16 compounds. The major compound was usinol. Usinyl acetate. This is followed by carfilene, and lastly, this was only alpha humulene, that is 2.75%. This is the image of the picture, sulfas and oil extracted. So harmful effects of synthetic pesticide, definitely ammonium phosphide can affect you if breathed in. Contact irritate the skin and eyes, nose, throat and lungs, causing coughing, wheezing and shortness of breath. Repeated exposure may damage the lungs, kidney and liver. Ammonium phosphide reacts with water and releases highly toxic and flammable phosgene gas. Definitely, other alternative is ethylene dibromide. This is liquid at ambient temperature. This can even cause irritation to skin, eye, mucous membrane, and upon breathing of lungs, liver, and kidney. These effects can result from any routes of exposure. The systematic effects of ethylene dibromide are in part due to metabolic conversion to the cell toxic and 2-bromoacetaldehyde. Persons with pre-existing skin disorder, eye problems, or impaired liver, kidney, or respiratory tract function may be susceptible to its effects. So that's why, because of its effects, a study was compared with sulfas and ethylene dibromide. So active oil here is clove, that is Syzygium aromaticum oil. Fumigant is again here sulfas, chemically ammonium phosphide, and other one is ethylene dibromide. Container used were tin container, and concentration here, this was 1000 ppm and 1500 ppm, and storage period was 60 days. Mycoflora of oil treated sample shows no fungus, that means complete protection. Sulfur aluminum sulfas treated sample shows agar plate and blotter. This is 6-4 at 1000 and 1500 ppm, and blotter technique it shows 6 and 3. Ethylene dibromide treated sample agar plate, here it showed 8 and 7 fungi respectively, in blotter 8 and 7, and control set was having 8. So here the techniques were both were used. A means agar and B means blotter technique. After 120 days of storage, again a study was done. Concentration even same, 1000 ppm and 1500 ppm, and storage period here is 120 days. After 120 days of storage, all treated samples showed no fungus, that means complete protection. Sulfas here treated showed 13, 11 respectively, 7, 5 respectively. Ethylene dibromide treated shows agar plate 13, 30, that means it's found completely found ineffective. So here control set 16 fungi or through agar plate and blotter techniques. So after this storage, hydrotoxic effect were studied. No effect on protein and carbohydrate content, seed germination, seedling growth, plant growth, foliage morphology, general health of plants. So again container was changed. This was we use gunning wax, and again the oil is clove. Container only changed, sulfas, and concentration is 1000 ppm and 1500 ppm. Storage period 60 days, and after 60 days of storage, oil treated shows no fungal infection. Sulfas treated says 7-4, and through agar plate, and 8-6 through blotter technique. Ethylene dibromide treated sample through agar plate 8-7, while at blotter technique it is 8-8 fungus, and control set was having 13 through agar and blotter techniques. A means agar and B means blotter. After 120 days storage in gunny wax, again concentration was 1000 ppm and 1500 ppm. Mycofloral analysis when done, clove oil was Syzygium aromaticum treated sample. This shows no fungus, complete protection. After 120 days of storage, ammonium phosphide, that means sulfas treated sample showed 10, 7 fungi respectively through agar, 8, 7 fungi respectively through blotter, and ethylene dibromide, this again found to completely ineffective. Control set was having 13 fungal species. So after 60 and 120 days of storage in gunny bags, no effect of seed, protein and carbohydrate content, percent seed germination, percent seedling growth, foliage, and general health of plant. So we may conclude the overall experimental work. These all can be exploited for preservation of peasant pea food source during storage due to its strong fungicidal nature, broad range of activity, capacity to withstand heavy inoculum density, strong repellent activity against pulse beetle, Calosobruchus chinensis, persistence of activity at high temperature, long storage period, doesn't affect seed protein and carbohydrate content, doesn't affect seedling growth, doesn't affect general health and plant morphology. So thanks to organizing authority for giving me chance to present my research work. Thanks a lot.
Thank you very much, Dr. Narendra Kumar, for your excellent presentation. So, any queries regarding this presentation, you are most welcome to ask Dr. Narendra Kumar so that he would be pleased to answer your queries. Okay, it seems there are no queries. Dr. Kumar, I would like to thank you once again for your presentation.
Thank you very much for giving me a chance to present my research work.
Yeah, you're most welcome. Thank you so much.
Next time, if you're doing any other conference, invite me, I will present again.
Of course, and sure, we will invite you for future webinars of United Research Forum as well. Thank you so much for your interest.
Okay, thank you from myself from Amity University, Haryana. My behalf and from Amity University, Haryana.
Yeah, thank you so much. We'll be in contact with you, okay? And so I would like to go for the next presenter, Dr. Bhaskara Phanindra. He was born in Chilamtur, mandal of Hindupur, Andhra Pradesh, graduated from Sri Venkateshwara Agriculture College, Tirupati, and post-graduated in Soil Science and Agriculture Chemistry from College of Agriculture, Bapatla, and PhD from College of Agriculture, Rajendranagar, Hyderabad. And is qualified agricultural research serviceman and joined as a scientist in soil survey and land use planning during 1919 at NBSS and LUP Nagpur. He is engaged in soil survey projects of Andhra Pradesh, Maharashtra, Kerala, the Tamil Nadu, and northeastern parts of country. He published over 150 research articles, one book, 12 survey reports, and presented over 40 articles in various seminars and served as reviewer to various journals. Dr. Bhaskara Phanindra, you're most welcome to start your presentation, and we are very excited to hear from you. Thank you so much.
Sir, are you listening me? Are you listening me?
Yes, we can hear you, but there is some sort of interference. Yeah, okay.
Now my slides are visible?
Oh, not yet. Can you share your slides?
Yeah, now we are sharing.
Okay.
Yeah. Okay. Yeah. Okay. Yeah. So thanks to the organizers. So my subject, my topic is on geo-environmental assessment. So in this geo-environmental assessment, the most of the work we are doing for land use aspects of major agroecological regions where the major crop production systems are really facing problem under dryland agriculture, rain-fed agriculture. So this is a project work assigned to me when I joined this regional center. So the land degradation is an important aspect where we are going to get a lot of information on abiotic stress, how it is affecting the crop productivity in rain-fed agriculture. Why the focus of rain-fed agriculture is more than 60% of cultivated land is under rain-fed, wherein the yields are almost stagnant and very low. And the yield per unit area is not improving much with our technological innovations in agriculture. So the main focus of geo-environmental assessment is on how we have to go ahead with sustainable intensification, and then how we can combat the climate change, especially in drylands. It is a very crucial issue for both soil science and also to the plant scientists. So in this particular part, I would like to quote one quotation. Where is the link between the soil and plant? Aldo Leopold. So he told that land is not the soil. It is the energy that is flowing in the circuits of soil, plant, and animals. So these three things are very much interlinked through our small holding farmers, especially under dryland agriculture. So with these points of introduction, I would like to say this is the topic of Pulivendala Tehsil of Kadapa district, which is a drought-prone area in Andhra Pradesh, where the crop failures are very common, and then the farmers are very resource poor. In this particular part, I would like to focus the next slide. So this is the abstract what I submitted in the optimized entire work of me in this particular presentation. The study area is a part of Kadapa Basin, which is having a lot of geological history, and wherein the geo-environmental issues are very high. The primary activity of this particular portion of Kadapa district is the mining activity. The mining activity is a primary occupation and agriculture is a secondary occupation in times of drought, where farmers are unable to get some livelihood from this part of the Kadapa Basin. So this Kadapa Basin is a sedimentary basin, and this particular study area that comes under a Papagni sub-basin of having a lot of geological variations, wherein the Pulivendala formations, Vemula formations of dolomite and limestone, and then some of the basaltic regions, and then quartzite hills of Gulcheru formations, and then the granitic landforms in the adjoining parts of the eastern parts of the Pulivendala. So the general soil types and land use, when you come to this particular part of the study area, so we'll see the sodic soils in the first soil and shallow soilness, and then the iron enriched, and then having a surface boulders. The stony soils are very high. And then the hill lands are mostly now they are converting into the banana cultivations under drip irrigation. And the majority of groundnut area, now it is replaced with banana because of government is supporting the drip irrigation with 98% of subsidies. So that is the major threat where the people are even shifting crop to the hilly areas, even under drought conditions. This is the ground realities existing in this particular part of study area. So looking into the problems of this particular area, now the stepwise activities generally what we follow in survey procedures, the standards are very much laid out. In that we have to go for 1:25,000 scale. The detailed, semi-detailed soil surveys we have carried out using both remote sensing data, that is Indian remote sensing satellite P6 with list 4 data sets merged with our soil survey topo sheets, and then we have to build up a geomorphological map of that particular area, where already I have slightly mentioned the 10 basic landform units were identified, and where we are going to the field survey for almost it took 3 years to do the soil survey of this particular area. this was coupled with the collection of climatic data and also the economic analysis, and then the evaluation of these land resource data sets as per the standards of FAO for banana and also irrigation. This is one of the most important activity in drought mitigation programs, and also the delineation of soil erosion risk zones, so that the priority areas for developmental programs where we have to focus. So this kind of activities, this is the how we have carried out this land resource informations. And I have here given only a few the climatic data sets, what we have done it. We have collected almost 109 years of rainfall data and analyzed it for the anomaly indices for rainfall and temperature. So in this, it is the mean air temperature, if you see the 30.7 to 36.9 degrees. And the rainfall is 679.59 mm with a coefficient of variation of 52%. So the temperature fluctuations are showing so much in this particular area, and the rainfall, extreme rainfall events earlier it is noticing very high up to 1960, and afterwards the rate of these extremely the humid periods are gradually reducing. And next we have done this one is Angot fluvial indices. When we worked it for decadal changes in this particular area, you can see that October and November. In the next slide, I will see. Yeah, this slide it will show you the normal and rainy and very rainy months are only confined to August to October, and then this is the period where interseasonal erosivity is very high in this particular part of the study area. So this is another the mean air temperatures of slide number 5, what I have shown it, the anomaly indices. When we see the mean air temperatures and developed a polynomial equation with the year, you see in the, from 1960s onwards, there is a gradual increase of temperature, because this area, it was very well recorded in the district gazettes that it is the place where the tigers are used to live in before independence. After independence, all the forests in the adjoining areas are destroyed, and there is no tiger at all in these areas, and that the loss of biodiversity that is clearly showing in this, how the temperatures are increasing in this particular area, followed by the rapid increase of mining activities in the adjoining hills. So when we relate this area on productivity of especially the groundnut growing area, this one is a major, after Anantapur district, Kadapa is a second highest groundnut growing area. When we worked that out for 1998, 99 to 2012. So the area also almost it will shifted when the rainfall is decreases. The season at which the rainfall decreases, the area under groundnut also it will decrease. And also the productivity, it is showing high fluctuations with the aberrations in the arrival of the southwest monsoon in this particular area. And this area, the rainfall is highly unpredictable and erratic. So after that, when we did, this is the climatic background of this area. And then we have derived a soil map as per the standards of United States of Department of Agriculture. In this, the geomorphological map we are going to develop and then identify the soil series through the ground truth. And then we have come out with 43 soil mapping units as soil series associations. So these are the baseline data that we are used for applying the Corine model to know what is the actual soil erosion risk zones when we follow the step-by-step the procedures outlined in Corine model. So in the actual erosional risk is very high in almost 66.96% of area in the particular tehsil. This is the region where we are all pumping so much of agricultural developmental activities, especially the high water demand crops. People are encouraging, and then they are going for digging so much of borewell. Irrigation is very high in this part of tehsil. So then we worked out the main indicators of the soils. When we say, see how much work one has to do to attain an optimal crop productivity. So generally we will take soil organic carbon stock. See, if you estimate for major series, so because the resource database is so valuable, it will show that it has worked out for 1 meter depth, all these major soils. So the carbon stock is very, very low in these particular soils. Whereas in some areas, the soil inorganic carbon, it is increasing. That indicates the degree of aridity in these particular areas. It is coinciding with the increase of carbonate in subsoils. So the next point, if I come down, the soil site suitability evaluations, whatever we are following as per FAO. There, the 42% of area is only suitable, which was very well marked in green. So these green belts are suitable for groundnut and the rest of area is not suitable. So the sustainable intensification is possible only 42%, whereas almost 80% of cultivated land is under groundnut. Yeah, then afterwards, when we work out the soil mapping unit-wise agroeconomic analysis, so there are only few units that are showing a net profit under optimum level of the input use. So the rest of the uneconomical portions are, we are seeing all these units are not economically profitable. We have to put more money and you have to harvest a little in this particular part. That's why the farmers are shifted to banana, because government is giving a lot of subsidy, 98% of subsidy for drip irrigation, and also for digging the borewell. So people have shifted to banana. Almost now the current land use is almost 22,000 hectares. From 2,000 to 22,000 hectares of land, now it is under banana, especially under irrigated system under dry land conditions, where the length of growing period is only less than 100 days. So this is the climatic water balance. Water balance is not supporting crop, but people are exploring the groundwater and also the canal water assured by the government from the Krishna River. This part almost, when we evaluate, that is coming to 38.33 with moderate limitations. This area is suitable for banana. Within that, again, if we worked it out, even if we worked it out, this drip irrigation is suitable for 43% of area. So this is how we have linked the analysis step by step for crop development and then productivity, especially the economic benefit and the rural livelihood, to the small farm holders in this particular region, and a small agroeconomic analysis for 3 major soil mapping units. It clearly shows that banana is fetching a 1.62 cost benefit ratio. 1 rupee for every 1 rupee spent, the benefit is 1.62 rupee. So these are some of the conclusions we have drawn, and this kind of analysis and this resource data, our organization is highly involved, and we are interpreting for the use of crop developmental activities, especially in dryland agriculture. So finally, I am concluding that I have to express my thanks to the Luis Anthony, the organizer of this meet, and asked me to present my results in this particular forum. This is only a case study how we are analyzing and using our land resource database for agricultural development at the ground level. Thanks to all the participants and the delegates for giving me this opportunity to express my sincere thanks to the organizers. So by this, I am concluding my lecture. So,
yeah, thank you.
Hello, sir.
Yeah, thank you very much, Dr. Bhaskar Final. Yeah, thank you, sir. Thank you very much for your nice presentation. Thank you very much. So any questions regarding this presentation, you can ask Dr. Bhaskar so that he would be pleased to answer your queries. Any questions? You're welcome.
Hello.
Yes, I would just allow the time for the participants. So if there are any questions, they will ask you.
These are the subjects that needs a lot of questions from the students. Yeah, because the regional case studies, very few case studies that are coming up, and then the resource experts are very little in this particular field.
Yes, exactly.
Yeah, because the soil landscape knowledge is very, very important for transferring your plant science knowledge.
Yes, there is a slight difference, and it's very interesting as well. Yeah.
Yeah.
Okay.
Because from the travel from lab to land, so a man has to face a lot of challenges.
Of course, Dr. Bhaskar. Yeah, you're right.
Yeah.
Okay. I would like to once again for your wonderful presentation, and I would like to move on for the next presenters, who is Professor Magdi Meher. Professor Magdi Mohammed completed his PhD from the Institute of Plant Breeding and Acclimation from Warsaw, Poland. He is the editor in the following international journals: number 1, DVT Journal of Agricultural Sciences, International Journal of Research, Granthalai, which is in Skupas, and International Journal of Applied Agricultural Research. He got the world's best teacher and world's best boss in biotrust systems for Africa International Workshop and World Agroforestry Center, Kenya. And he's also major participant in the varieties invention of some forage crops. And Professor Magdi Meher, you're most welcome to begin your presentation, and we are very excited to hear from you. Thank you. Can you please unmute your microphone? We are unable to hear you. Can you please unmute your microphone? We are not audible. Your microphone is, yes. Yeah, hello. Can you speak something so that I can change?
Hello?
Yes, yes, it's perfect now. Professor Magdi Meher, you can start now, yeah.
Can share my presentation, sir?
Yes, you can share your presentation now. And Professor, is there any problem in sharing your slides so that I can guide you regarding that?
I have some problem. I have some.
Oh, okay. Okay. So, did you send your presentation to my colleague Luis?
Yes, 2 days ago.
Okay. Let just me check with that.
Yes, sir.
Yeah, Professor, is that your presentation, right?
Yes.
Professor, is that okay if I go with the next presenter, Rabia from Egypt, and can you present in the next after Rabia? So that meanwhile I can load your presentation. So I will take it from my colleague. Is that okay for you?
Okay, okay, Professor, as you would like.
Okay. So I will ask Dr. Rabia, the next presenter. Dr. Rabia, are you ready to present now? Is it okay for you? Your microphone is muted. Just can you please unmute your microphone?
Yes, how can I share my presentation, please, share?
Okay, you'll find a share screen button on the bottom. Can you see that on your screen?
Yeah.
Please click on that share screen.
Yeah.
Okay, and it asks to share your exact PPT that you're going to present now. So please click on share screen and load your PPT there so that you can share your screen.
On Windows or Chrome tab?
It is in Windows. Click on Windows. Okay. Did you open your PPT first? I mean your presentation. You need to open your. You need to click on your presentation first, and then you need to share screen.
Yes. Okay. It's opening now.
Not yet. Maybe still loading. I think so. So I just wanted to tell you that. So please open your presentation first, and then you can click on share screen.
opening now.
Okay, let me check with that. Yes, all right, that's perfect now. We can see your presentation, right?
Okay. This is the beginning.
Yes, yes, we can see your presentation. You can please go ahead.
My name is Rawia Omar Shamsuddin, working in ARC Egypt. My presentation today about the effects of stress-surviving microorganisms in wheat and spaghetti. You are hearing me?
Dr. Ravia, we can hear some echoes from you. I think you just log in from two devices, so there's some sort of interference occurring. There is some echo that we can hear. We can hear you, but there is some sort of echo. Can you just speak something out so that?
You're seeing my presentation now?
Yes, yes, we can see your presentation. That's perfect, but we can see, we can hear echo as well. So we can.
This is the beginning?
Yeah, okay. It is somewhat better. You can start now.
Sorry, I lost my presentation. Okay. Okay. My presentation about effect of stress-surviving microorganisms on wheat yield and phosphate activity. We know the problem in phosphorus in the soil that it most of 100% of phosphorus become mobile and unavailable.
Dr. Avia, I'm sorry to interrupt you in the middle. I can see just a white screen. I cannot see your presentation. I can just only see a white screen.
I shared my presentation now.
Yes, I think you have shared your presentation, but we can only see a white screen here. Can you please stop your screen share and reshare it once again so that it will be right now?
Okay, sir.
Yeah, okay. Yes, now we can see your screen.
Okay.
Yes, we can see your screen. You can continue now.
Bad internet network. Sorry, sir. مش عارفة أعمل من هنا. مش واقفة هنا. So open now.
Yes, we can hear you now.
The problem of phosphorus in the soil, that the most of phosphorus becomes immobile and unavailable for plant. There is a lot of phosphorus in the soil, but it becomes
fixation and absorption.
it's dependent on soil type and pH. The problem is that the most of Egyptian soils are rich in phosphorus, but it most of insoluble form. Only less than 10%
of soil phosphorus uptake in the plant animal cycle. We know the role of phosphorus, many numerous rules in sorry. I can see the presentation.
It's okay. We can hear you. Please continue. We can hear you well.
You can see my presentation.
Yes, we can see your presentation and we can hear you. No worries. You can start. You can start your presentation.
Okay.
Yeah.
The role of phosphorus in the soil. We have numerous rules in the soil of phosphorus that we can know. It promotes seed production and increases stem strength. Phosphorus plays a significant role in plant maturity and growth and development. Okay.
Please follow.
The world direction in the agriculture that organic agriculture by using biological control and biofertilizer. The biofertilizers like phosphate-solubilizing microorganisms and fungi like arbuscular mycorrhiza fungi, we can use these strains from isolate phosphate-solubilizing bacteria and fungi to improving the availability and solubilization of phosphorus in the soil. So the rationale of the study was to have make the phosphorus more availability in the soil and be soluble more than in the following, and decreasing the using of chemical phosphate fertilizer that it has a negative impact of soil consortia and decreasing the cost of agriculture farm. Sorry, I can't see my presentation.
We can see your presentation. Dr. Rabia, we are glad to see your presentation. We can see that. So at present, we can see the slide regarding the lab experiment.
Yeah.
Yeah, you can continue, yeah.
أنا مش عارفة اللاب والفون الاثنين داخلين في بعض. ما هي كده الفون هو اللي واضح.
Dr. Rabia,
Slide number 8.
no, we cannot see it now.
You can see the slide number 8.
No. It's we cannot see your screen at present. So, do you want me to share your PPT?
Yes.
Is it okay? Okay. Just a moment. I will do that for you. Okay, is the one you're presenting now?
Yes.
Okay, you can speak now so that I can move the slides.
Okay. The objective of my study was to evaluate the effect of phosphate-stabilizing bacteria alone or in combination with arbuscular mycorrhizal fungi to increase the availability of soil phosphorus in wheat yield, and then to evaluate the effects of this bacterial inoculation on activity of soil enzyme to obtain healthy food by using microbial inoculants for the economic crop such as wheat. We used two experiments. Firstly, lab experiment. We use rock phosphate to estimate the rock phosphate solubilization in liquid culture. Our culture uses that the National Botanical Research Institute phosphate broth media, and triplicated samples were taken after 0, 2, 3, 7, 10, 11, and 14 days post-inoculation. The culture suspension were centrifuged and estimated pH in the supernatant in regular intervals. The experimental design, it was the second experiment. The design was a split bulb design. The estimated after plant harvested vegetative stage and plant harvested at ripeness stage. We was have 20 treatment in 3 replication. The main bots in treatments was 0 and 31 kilogram P2O5 per feed, and the subspots was arbuscular mycorrhizal fungi, non-inoculated and inoculated. Subbots was 4-3 isolates from bacteria, and the control was non-inoculated. Soil and the plant analysis, we take the available inorganic phosphorus and pH in soil chemical analysis, but we take activity of alkaline phosphatase in biological analysis. In plant analysis, we estimated nitrogen, phosphorus, potassium concentration. Collected data, we take the show dry weight and the green dry weight, straw yield, and biological yield. In this table, we can see that the soil was sandy soil in pH high, more than alkalinity. The soil was very poor in nutrients. In first experiment, we showed that the result in this experiment indicated that the level of phosphorus related from rock phosphate increased with longer incubation period, and the amounts of phosphorus released into the medium was dependent on the type of strain. In the second figure, we can see the relationship between phosphate solubilization and pH in power function, indicating that the response of phosphate solubilization is proportional to the explanatory pH raised to a power. The table of the data was in this table the effects of rock phosphate and bacterial strains in interaction between arbuscular mycorrhiza fungi on the soil available phosphorus and pH in weight after 2 duration 69 days and 130 days in vegetative and ripeness stage. In this table, we can see that the soil available phosphorus was significantly increased by inoculating the soil and arbuscular mycorrhiza fungi and phosphate-solubilizing bacteria. Some available phosphorus was significantly increased comparing with both of non-inoculated or individual inoculated in soil. In this table, we estimated the effects of rock phosphate and bacterial strains interaction with fungi on the activity of alkaline phosphatase in 2 duration in vegetative and ripeness for wheat sowing. We can regarding the interaction effects of rock phosphate application of arbuscular mycorrhiza fungi and phosphate-solubilizing bacteria inoculation on alkaline phosphatase activity on soil. The alkaline phosphatase activity was significantly increased by inoculating the soil with arbuscular mycorrhiza fungi and phosphate-solubilizing bacteria, comparing with both the non-inoculated and the individually inoculated soil. Applying rock phosphate at the rate of 31 kilogram P2O5 per feet to arbuscular mycorrhiza fungi and phosphate-solubilizing bacteria inoculated soil resulted in a significant increase in alkaline phosphatase compared with the untreated rock phosphate, arbuscular mycorrhiza fungi, and phosphate-solubilizing bacteria non-inoculated soil. Alkaline phosphatase was higher after 69 days than those after 130 days from wheat sowing. In this table, that's the correlation coefficient between soil pH values and available phosphorus, alkaline phosphatase in rhizosphere of wheat plants in 2 duration after 69 days and at ripeness. For wheat plant, the reduction in soil pH due to the organic acids produced by arbuscular mycorrhiza fungi and phosphate-solubilizing bacteria inoculation are responsible for the increasing of available phosphorus and alkaline phosphatase activity in soil. So in this table, the relationship between rock phosphate, bacterial strains, and arbuscular mycorrhiza fungi on plant growth and yield in 2 duration in vegetative and ripeness for plant. Applying rock phosphate at rate of 31 kilogram P2O5 per feet to arbuscular mycorrhiza fungi and phosphate-solubilizing bacteria inoculated soil resulted in a significant increase in shoot dry weight compared with the untreated soil. With rock phosphate, arbuscular mycorrhiza fungi, and non-phosphate bacteria. Non-inoculated soil after 69 days from wheat sowing. The effect of rock phosphate, bacterial strains, and arbuscular mycorrhiza fungi on uptake of nitrogen, phosphorus, and potassium in shoot of plants, wheat plants after 69 days from sowing we can see that the concentration and the uptake of NB and K in wheat shoot samples after 69 days were significantly increased as a result of the individual rock phosphate application and arbuscular mycorrhizal fungi and phosphate-soluble bacteria inoculation as compared with the non-treated soil. Generally, applying the rock phosphate to arbuscular mycorrhizal fungi and phosphate-soluble bacteria inoculated soil was significantly increased the concentration and the uptake of NPK and K in shoot wheat samples, comparing with rock phosphate untreated soil. Phosphate solubilizing bacteria strains, Pseudomonas fluorescens, Bacillus megatherium, and Serratia marcensis were more effective in increasing the concentration and the uptake of NP and K of wheat shoot and compared with Bacillus subtilis soil amended with rock phosphate and inoculated with arbuscular mycorrhizal fungi. Conclusion: the green straw and biological yield were significantly increased with the application of phosphate-solubilizing bacteria and arbuscular mycorrhizal fungi as a result of its positive effect on the release of phosphorus from rock phosphate and transform it into soluble from the adsorption of plant, which reduces the application of phosphatic fertilizers and therefore reduces production costs. Therefore, Egyptian farmers can use these microbial inoculants as biofertilizer with rock phosphate for wheat growing in sandy soil. It improves soil fertility and decreasing the costs of agriculture. However, this study needs to be estimated in the field condition under different soils and environmental conditions. Acknowledgement to Soil and Water Department of Faculty of Agriculture, Suez Canal University. And I would like to thank my supervisor in Soil Water Environmental Research Institute. Thank you, my husband, for helping me. Thank you for conference coordinator.
Thank you very much, Dr. Avia, for this presentation. Do you have any questions to discuss with?
Hello, good afternoon, everyone.
Yeah, very good afternoon, Dr. Oman Bernard.
Hello, good afternoon.
Yeah, hello.
Yes, I just have a comment, question. Yeah, thank you very much for your presentation. I just wanted to ask, you use the isolates, individual inoculation of the isolates. Did you think of having a consortium of microorganisms? Because from literature, we find out that using a consortium of microorganisms gives better results than single inoculation of the microorganisms. And that will involve carrying out antagonistic experiment in vitro to see if the isolates are compatible with each other before they can be combined for inoculation. Then I think this is a greenhouse experiment. Like you said, you are going to go to the field. But one thing I've noticed is that the situation in the field, it might do very well in vitro or in the greenhouse, but when you go to field conditions, the isolate might not be strong enough to compete actively with the indigenous microflora. I think you should also consider that before you go to the field. Thank you.
So I think firstly, you asked about the combination with arbuscular mycorrhizal fungi. You mean that?
Yeah, I'm asking if you considered using the consortium of the microorganisms used. For example, Pseudomonas fluorescens did very well. If you combine it with one or the other microbes, any of the other bacteria, it could give you a better result than individual inoculation.
You hear me?
Yes, I can hear you.
Okay, I combined everyone isolates with arbuscular mycorrhizal fungi, not the 3 isolates with arbuscular and another. Okay, the second question,
Thank
please.
You. Okay. Yeah, the second question is just a comment that when you move from the greenhouse or in vitro to the field, the situation is likely to be different because the microbes might not be able to compete with the indigenous microorganisms under field conditions because you did it under a controlled environment. So going out to the field, you should expect something a bit different. Just a comment.
Hello.
Sorry,
Hello.
again, please. Sorry,
yeah. So, I would like to know how much area is under sandy soils? How much area you have under sandy soils in Egypt?
The second experiment was in bots, in 25 kilogram bot.
No, because why you have selected sandy soils for phosphorus?
Again, please.
Yeah, why you have selected sandy soils for phosphorus studies?
Because most of Egyptian soils are sandy soils.
So how much area it is?
In Egypt?
Yeah.
More than 100%.
Okay, okay.
Thank you.
Thank you for your nice presentation. Thank you for your nice presentation.
Professor. Thank you, my professor.
Okay.
Yeah, thank you very much, Dr. Rabia, once again. So, and I would like to thank you, that's it. So let's move on for the next presentation. So the previous one, Professor Magdi Meher. Professor Magdi Meher, are you ready to present now? Yes, and I would like to request you to please unmute your microphone. Your microphone is muted. I would like to request you to unmute that.
Okay, Professor.
Yeah, okay.
Our presentation about the strategic study on novel technology approaches for transforming agriculture and industry bio-waste into bioproduct in Egypt. This work part from Egyptian Finnish project. In Egypt, agriculture and industry practice produce a large amount of wastes. Bioreactor system is a novel technology approaches for transforming this agriculture and industry bio-waste into bioproduct. For that, to maximize the benefit and the return from this waste. The problem we have in Egypt about 55 million ton bio-waste. This statistics for 2020. This bio-waste is blending of snake, squirrel rates, mosquito and flies, especially stable fly, which bleeded the human and the animal in Egypt. On the other hand, investigations explored in Egyptian destroyed potential feed stock for value-added product with local application. The opportunity, we assess new biotechnology methods, conserve of bio-waste by biorized system, cost benefit analysis of this new technology adapted to social, economic, and environmental conditions in Egypt. These retro machines exported to Egypt from Finland, and these machines, the capacity for these machines only quarter ton for the experiment work. It is also to mention that only in the Africa and in Asia, these machines in North China and Egypt. That's the only. We have some, we do many cycles from many wastes, as we explain after. This some liquid or some tar for machines, and we see the gases and also the biochar. The biomass or the agriculture industrial waste, we do biorizing system. We product the biochar, three types: biochar, bio-oil, and gases. The percent for this component was different from the slow polarized system or fast biorized system. In slow polarized system, the product distribution third by third. For example, liquid 30%, gases 35%, and biochar 35%. In addition, in contrast, in case of fast polarizes, we obtained 75% liquid, 13% gases, and 12 biochar only. The output is divided to three components. Non-condensed gas, this, we can use this as energy. Condensed gas, when the machines started from 0 to 300 centigrade, the hemicellulose we will be polarized, and we obtained the liquid as a herbicide. From 300 to 400, the cellulose will be polarized, and in this case obtained the insecticide. More than 400, the lignin will be biorized, and we obtained fungicide. The biochar system, the biochar is only as a soil amendment, not fertilizer. In this case, we have some technique to obtain the biochar as a soil amendment in the sand soil or clay soil. In addition to, we must be added some fertilizer. But when we used from our experiment in North China, the sand soil, we obtained the good result as a soil amendment. We can save more than 35% from water requirement for many crops in Egypt. In this case, we can increase the cultivated area, which we need, especially as we know now, we have problems from Nile River with Ethiopia. In this case, how we can keep the water around the plant, and we save the water. The The source of agriculture and industrial waste, seasonal bring of free, especially olivia, citrus, peaches, mango, and date palm, vegetable crops, tomato, watermelon, pea, onion, potato, pepper, and eggplant. Sorry. Field crops waste, especially rice, barley, wheat, lentil, corn, faba bean, sugar beet, sugar cane, and canola. Industry waste from olive, tomato, citrus, sugar beet, sugar cane, and date industry. For the market, this technology, we total cultivated area about 7.2 million faddan. It is equal 3 million hectare. Total cropped area annually about 13.3 million faddan. This equal 5.6 hectare. Total animal weight in Egypt about 20.4 million heads. Now we have several shortage of water resource, and is therefore in urgent need to applying new technology standard to maximize the utilization of available water, such as our mentioned proposal for saving water. Now, the market and offering this new technology described in this figure, we can from the soil or the valuable sustainable, we can soil benefit from decrease leaching water and fertilizer, improving soil fertility, improving soil health. For biochar market now, we use or the world use biochar I am very sorry because now we in Ramadan months and we fast, and don't apply for me to drink water or anything. I am very sorry.
It's okay, Professor. No worries. It's okay.
We do many bioreactor cycles for many bio-waste, as mentioned in this table. Some of them in Egypt and the other in Finland cycle. This liquid or the tar product for herbicide, fungicide, and insecticide. And when the temperature reaches to 750, we can use a tar as a paint of whole. This is some result from our cycle in Egypt, but it is we can see all the summarized in the first table. Okay, sir. This average of 30 cycles of tomato straw. This average of 12 cycles of palm waste in North Sinai and Egypt. We can see the right this condensed gas and to obtain the liquid at many different temperature. On the left side, we can see the energy. This is used as energy. Gas is produced from these machines as energy. this is a powerful for bioinsecticide against palmweed. We can use as this when we obtain the liquid at 270 to 350 centigrade. As we see in the left pictures, how we can use the gases as an energy source. The tar product liquid from tomato straw is best one as a herbicide, especially on field bindweed. This weed to do many problems in agriculture in North China. Some practicals, we added the biochar as a soil amendment. This for palm tree. The competition for this technique, we have, we can use all this waste as a compost. But when we see some calculation or some estimated, when we use the old technique that we do as a compost, we can, as we see, from 55 million ton, we can obtain 40 million ton as a compost, as a percent of 25.5% as a dry matter. By the compost obtain about 300 Egyptian pound, you can see how many we can obtain from the old technique. But in the right, the new technique polarized system, we obtained 3 components. The first one is biochar, second biopesticide, and until now unknown gas volume. We can compare what benefit from this new technique from this slide. Okay, sir. Business model, we can selling or market the technology, or we can do some national project or business sector process. We can do sell product directly or partnership with other company. Some product I am seeing this in Italy when we have a meeting in Milano. The biochar used as a cakes and also make a bread from the biochar. Okay, sir. Biochar as a human food, as we explained. Okay, sir. For animal feed, to reduction of hydrogen. We also use in the breeding chickpea in the natural environment of biochar, as we see. I am published this work for 3 international conference, and we obtained the best world in teacher and the best world in post for management this work in Egypt. This is some work for my professor and my colleagues. We can see to review this website from our activity from this target area. And thank you so much. And I am very sorry for what happens.
Yeah, it's okay, no worries, the professor, and thank you very much for this excellent presentation. And in fact, that's very interesting that you has been awarded as a world's best teacher and world's best big boss. And thank you so much. Any questions regarding this PPT from the audience? Or else you can type your queries in the open chat box available at the bottom so that I can address your queries to Professor Magdi.
Thank you, sir. Thank you so much, sir.
Yeah, you're most welcome. So on, without any further ado, we will move on for the last presenter for today, Dr. Omina Bernard Azuriri. Okay, before that, I'd like to have a small introduction.
Good afternoon.
Yeah, Dr. Omina, so I'd like to introduce about you and then you can start your presentation. Yeah, Dr. Omina is a senior lecturer in the Department of Biological Sciences at King's University, Nigeria, and an extraordinary senior lecturer of the Food Security and Safety Niche area of the Northwest University, South Africa, and part-time lecturer at the Pan-African University Institute of Life Sciences, including Health and Agriculture, Ibadan. He holds a BSc Honors in Microbiology from the University of Benin, Nigeria, while his MSc in Environmental Biology and PhD in Plant Genetics were obtained from the University of Ibadan. He is a recipient of International Foundation for Service, IFS. Dr. Omina, you're most welcome to begin your presentation now, whenever you want. Thank you. Dr. Omina, can you hear us? Can you hear me? Dr. Omina, we cannot hear you. Is there any technical issue regarding that so that I can help you? So I would like to request the audience to cooperate for just a couple of minutes because I think there is a network connection with Dr. Omina. So he was not in the Zoom meeting, so he just left out. Maybe he can join us within a couple of minutes. So I would like to request you to please be with patience so that we will let him within 1 to 2 minutes. Okay. Thank you for your cooperation. So on this last presentation for today. So in the meanwhile, so any one of you are welcome to discuss about your research that is happening with your university or with your colleagues. So you're most welcome. In the meanwhile, Dr. Omina will join us. So our team is calling Dr. Omina. So maybe he's facing some network issues. So I would like to request to all the audience to be wait for a couple of minutes because he's facing some network issues. So our team is calling him, and this will be the last presentation for today. So just a couple of minutes. We will wait for him. And Dr. Ivana, this is the last presentation for today that Dr. Omina Bernard, so he's the last presenter for today. So in the meanwhile, so if the participants are interested to share your research so that is happening with you or your colleagues that you're going to do in the university, so in the meanwhile, you can share with us. So you can speak something out and you can come forward and you can speak something out regarding that. So there is a feedback link provided by my colleague Luis Anthony. So you can comment, you can post a review. So you can click on this link. So it can so that redirects to a Google form. So where you can write your comments there. So everyone in the Zoom, you can please click on that link. So it redirects to a Google form link. So and then you can post your feedback about that, about UNITRO Research Forum. So how do you feel about this webinar? Okay, and maybe so we will end the presentations now. Maybe Dr. Omina Bernard is not available, so he's facing some technical issues, and we will end the webinar for today. And before that, I would like to poll some questions that will be displayed on the screen right now here. So I would like to request all the participants and pay kind attention so that we will take a feedback from you and we will review ourselves regarding the organization of our future webinars. And here I would like to launch the first poll question. So I think, so can anyone acknowledge me with yes or no that you can see my questions on the screen? Or as you can type in the chat box that you can see the questions on the screen. Yes, thank you for answering. And here I would like to go with the second one. Thank you so much. Yeah, yes, we could see them. Thank you so much for acknowledgement. So here I would like to go for the fourth. Thank you all for your positive response. So here is the last poll question for you. Yes, thank you all, and thank you very much for your response as well as answering the questions. So, and I would like to thank one and all the speakers who have participated in this webinar, taking the time apart from the busy schedules. So I would like to thank by name Dr. Walid Fawad Abotar from Egypt, and Dr. Ivana Sola from Croatia, and Dr. Zogan Ko from South Korea, Dr. Irin Devisi from Greece, and Dr. Alam Hassan from Egypt, and Dr. Yusuf Mohammed from Egypt, and Carolina from Lithuania, and Dr. Iqbal Hussain from Pakistan, and Dr. B.N. Hazarika from India, and Shweta Gupta from India, Narendra Kumar from India, and Bhaskara Fanindra from India, and Mike D. Maher, Professor Mike D. Maher from Egypt, and Dr. Rabia Oshams from Egypt, and unfortunately, Dr. Oman Bernard is not here because he's facing some technical issues. But however, I would like to also thank him. And so I would like to hand over my mic to Dr. Ivana Sola from Croatia, so that she would be closing the conference with a small closing speech. Dr. Ivana Sola, you're most welcome to share your thoughts with the participants.
So yes, thanks for the words you gave me. I want to thank all of you for participating in this webinar. It was really interesting. Some of the presentations were more close to some of us because they're more similar to what we are searching for. Some are a bit different from what we are working in the lab, but it's always nice to hear something new, something that's it's not so familiar to you. So I think it was really nicely organized. We got a lot of nice informations, probably some future collaborations as well. Yeah, I would like to thank you all of the participants for sharing their results, ideas, yeah, and I hope we will see us soon at a similar way. Yeah.
Yeah, thank you very much, Dr. Ivana, for your nice closing speech. So is there any from the speakers that you have presented today that you want to thank the remaining participants from across the world, because we can see the participants from across the globe here. So if you can share your thoughts with us, that would be great.
Hello.
Yes, Doctor. I can see Dr. BP Bhaskar is speaking, right?
Sir, I am very much thankful to the organizers. So I am a odd man out because we are all, I am working in the field of geosciences, and with a particular reference to your plant environment. So this forum has provided me an opportunity to know what is the exact place of the kind of work what we are doing at a regional level issues, especially for plant developmental activities. This is a very crucial point here. How this energy flow, it is flowing from soil, plant and animals. And the issues are very critical, especially in the dryland agricultural systems. Unless people will come together to make use of the land resource databases and then the resource maps, what we are generating, and also the guidelines what we are following for plant adaptation strategies coupled with conservation planning. It is very, very important. If we are interested only in crop development, I think we are ignoring the conservation planning of the land resources, which is a basic material for any developmental activities. So this forum has provided me to know what is my position, and I have come across some of the important presentations that add something to widening my wisdom and also the knowledge. Yeah? So I am very much thankful for the organizers for giving me this opportunity, and also in meeting all the people who participated and presented very good material. And also I thanks to all participants on this occasion for patiently listening all the presentations. Thank you. Thank to the organizers.
Thank you very much, Dr. Basu, for your nice words in your presentation. It's not an out one out, but there is a relation between the soil as well as plant science. But your presentation is very interesting as well. Thank you. So, is there any participant or any speaker that you want to speak out and come forward and speak out? You're most welcome. So we'll be closing within a couple of minutes.
Hello?
Okay, I think there are no more to speak. Okay, it's okay, no worries. And I would like to thank you once again, my director of organization, Dr. Ranga, for providing me this opportunity to be serving as a moderator for the United Research Forum conferences. So as well, so if you are interested to collaborate with us, or if you want to present a paper or submit a paper and to want to present at the international conferences at United Research Forum, so you can contact me via email at john@unitedresearchforum.com, or you can WhatsApp me at +44 744 880 8243. So you can contact me so that we can have a collaboration and we can have some discussions, positive discussions regarding your research. So, and thank you once again all for joining with us for today's webinar. So, and hopefully I would like to see you in the future webinars of United Research Forum as well. And thank you, and have a good morning or good afternoon or good evening or good night. And I would like to thank once again Dr. Ivana Sola from Croatia for accepting as soon as to have some role as a training session as well. Thank you so much, Dr. Ivana Sola, for it.
You're welcome.
Yeah, thank you, Professor Magdi. Yeah, have a great day. And Dr. Iqbal Hussain, have a great day. And I can see Dr. Irene Devsi, have a great day. Yeah, and Dr. Carolina and Dr. Yusuf Mohammed and Dr. B.N. Hazarika, Dr. Shweta Gupta and Dr. Narendra Kumar and Dr. Bhaskara Fanindra and Professor Magdi and Dr. Ravia and Dr. Omina Bernardo. Thank you all. Thank you once again. Have a great day. Yeah, have a great day. Take care. Bye-bye. See you all. Yeah.